Welcome to Translation 2024. We have a fantastic program for you. It's already started off with the Early Career Researchers program and the Trials Delivery program this morning. This is the main conference, and we have speakers from all over the world, actually, apart from our sort of UK speakers. We have Charlotte from the Netherlands, and I'm so pleased that you could join us, Charlotte, and Do-Geun from South Korea, so that's wonderful. Ola from Norway, Nick from Sweden, David from the USA, Stephen from Germany. It truly is an international setup that we have here. But I'll tell you what's even more interesting. There are so many new faces, and I cannot tell the industry from the academics. And it's just that lovely sense of we're all in this together to try and solve the issues surrounding dementia and dementia translation. Now, I must do some housekeeping. And so I've been told I must do some housekeeping by my team. And as a man under authority, I always do what my team says. So first of all, and this is not onerous at all, I would really like to thank our sponsors. Here they are along the bottom. We have the Alzheimer's Disease Data Initiative, Acrivia Health, Alzheimer's Research UK, Alzheimer's Society, ASI, Lilly and Roche. And they make this conference possible. And so we remain extremely grateful to them, but actually really pleased that they wish to partner with us in bringing both the academic and the industry sector and the third sector together. I think the bigger context for this is that we have been invited by MRC to renew, and this doesn't mean to say we get a pass, far from it. We go through exactly the same, if not more rigorous review, peer review, than other projects because of the scale and the complexity of who we are. But we are in active renewal preparation mode, and if you would like to be part of that renewal, if you think you have something to contribute, which will contribute to the field as well as to who DPUK is, we would like to hear from you. And so part of what we're doing today is saying, this is who we are, this is what we do, and if you want to contribute, you are really, really welcome. To give you a flavor of what's coming up, basically DPUK has three main areas. We have our data portal, which I think is one of the most mature and sophisticated secure data environments on the planet, actually. I think barring GCHQ and other government agencies, I think that it's really a first-rate world-leading data portal. We have a national trials delivery framework, which has registers, the largest registers in the UK, 68,000 people signed up for recontact for dementia trials, and a network of many sites, including primary and secondary care, as well as just our preclinical registers. So it really does offer an opportunity for people to sign up for trials. And we have our experimental medicine incubator, which I think this is possibly of most immediate interest to industry, because what we do is effectively co-design with industry partners what are the questions that you are asking and how can we help you answer them. And the idea is to de-risk go, no-go decisions for very expensive clinical trials. And so reduce screen failure, reduce trial failure, and accelerate the development of new treatments. So that's what we do. And what this does is give you an indication of the level of activity. I'll just mention the data portal briefly. We have 1,000 users in 50 countries. We have processed over 1,500 data access requests. And we have a median access data request response time of, which means yes or no, with qualifications, of 22 days. And I think that is remarkable. And if any of you have been involved in data access requests as a third party, you will realize that this is an extraordinary level of service delivery. And so we have other things as well. So that's really who we are. And the beauty of this is that my job is now done. And I'm going to hand over to James, who's our Associate Director for Experimental Medicine, and he's going to chair the rest of this session. James.
Welcome to this first day, the first session of the Translation 2024. Whether you're in industry or academia at an early career stage or mid-career stage or established career stage, or in the third sector, whether you're patient representative or clinician, it's a very warm welcome. You will almost certainly know some people, but I bet there's nobody that knows everybody in the room. So around your table, make sure you talk to the person on your left and the right. At the tea breaks, talk to the person you've not met before. And it'll make it an even more exciting event today and tomorrow. This afternoon, we'll talk about biomarkers, and I'll reflect on biomarkers for a few moments before introducing our speakers for the afternoon. And that will lead into discussion tomorrow around big data, what it means, how to do it, how to get it right, and trials and trial delivery. So when Murray walks down Sesame Street, okay, and has since for 50 years now, he asks everybody he meets what they think the word of the day means. Okay. And I don't think he's asked what biomarkers mean, but I'd love to know what answers he'd hear. I think as he walked down the street, he'd get many different answers. Some people might have read the FDA and the NIH sort of overview and guidance booklet. But if ever there was a word in need of some sort of adjectival enrichment, some explanatory enrichment, it's biomarkers. And there's been a lot of confusion, I think time and effort misplaced because of a lack of clarity over whether the biomarkers that we're thinking and talking about are for diagnostics, are they for prediction and prognosis, are they related to safety and risk, or their role in monitoring or response assessment. These are different properties that biomarkers might have, So if we think what the biomarkers are, well, we'll hear a lot today about blood, but we should remember it's not the only type of biomarker. Blood has many advantages for scalability, but we can also image the brain with PET or MRI. We can look at the eye through optical imaging. We can look at other biofluids, typically CSF, but also we shouldn't forget what saliva and urine can add. There's cognitive tests, neurophysiology, EEG, MEG, and digital technologies coming to the fore increasingly. All of these might individually or jointly develop biomarkers, and a lot of emphasis then needs to be put on how we bring data together and that sort of computational overlay. Whether it's AI of the moment or more simpler traditional techniques, it's how we use and think about the data. One of the questions is to compare these types of biomarkers. And one of the DPUK studies that's now well underway, will be drawn to a close over this next year, is the deep and frequent phenotyping study led by Vanessa Raymond, set up by Simon Lovester, now run by Vanessa Raymond, which essentially pits these methods head to head. Okay. All of these in the same subjects frequently over the course of a year to look at their relative properties and understand their relationships. I sometimes think the strapline for this could be, what is the best and fastest way to know if a drug works, or if it doesn't work, can we discontinue it, working against dementia, head to head. But we'll hear today a lot about blood biomarkers, so it's particularly exciting that now, as of the last week or two, we can now talk about and discuss the new and exciting project funded by the Alzheimer's Research UK, Alzheimer's Society, the Postcode Lottery and the NIHR for a multi-site pan-UK-wide study called READOUT, Real-World Dementia Outcomes. This is led by Vanessa Raymond, supported by myself and Iain. It's a major investment in blood-based biomarkers to start this summer and run over the next 5 years. Vanessa will talk tomorrow in the trial section a little bit more about the structure and design of the overall study, but I wanted to touch for a few minutes now on how the blood-based biomarkers fit in and what the study comprises. And essentially, we'll be looking for 3,000 volunteers through memory assessment services, secondary care, and we'll be measuring in blood a range of markers. Now, I've written up here what's called tier 1 markers. These are biochemicals that are either already in use in different parts of the world or clearly ready for consumer and healthcare use. We hear a lot about 217 as an example, phosphatide 217, but there are others that are sort of oven ready for use now through healthcare systems, with some caveats. There's a range of blood tests that have extensive research data in large cohorts, often longitudinal, but then they need some more technical expertise, or they're slightly less developed in their advantages and disadvantages and risk mitigations. but they are well understood and used at scale. So for example, some of the cytokine panels or GFAP might come into this group, and others. And there's tier 3, really exploratory biomarkers, and I'll come back to those in a little more detail. What we'll be asking is across the range of dementias, not just Alzheimer's disease, and not just Alzheimer's dementia versus healthy controls, but the differential diagnosis amongst all those types of people that come to memory assessment services, whatever the diagnosis. How might these biochemicals in blood be used on their own as a singular marker, I'll put it univariate, a standalone marker? Something perhaps like glucose and diabetes, on its own as a single biochemical, it can be highly informative. Perhaps a multivariate, so a combination of markers, the combination of biochemicals, as indeed we do in other branches of medicine. We use a lipid panel commonly, not frightened of having lipids. As a set of HDL, LDL, cholesterol, we use a combination of biochemicals together. Or anyone who's using CSF biomarkers now, you get a panel of 3 or 4 different biomarkers. These might be enhanced further by combining with relatively simple clinical and demographic information. It's a little bit more complicated, but already the type of thing in standard practice, such as ABCD score for a TIA stroke risk assessment, combination of things that you measure and some demographics of the patient, already in regular use day in, day out. There may be some additional work for more advanced AI algorithms, more sort of black box methods to really improve performance, but then really with the challenge around generalization and trust and understanding that comes with that. But for any of the biomarkers, whether on their own or in combination, we'll be wanting really the best performance. Now, performance you might think of as accuracy. How accurate is it? And that's intuitive understanding of the quality of a biomarker. But actually, we need to think more about positive and negative predictive value. How useful might that be for an individual patient, either in clinic or as a prospective trial candidate? So positive and negative predictive value. And that speaks to sensitivity and specificity against the background prevalence of different disorders. The decisions about how biomarkers behave, what their thresholds are, what are the pros and cons of false positives and false negatives, what's acceptable to patients and the public, what's acceptable to healthcare providers, that will also be a very important perspective. So it's not just in the statistics, it's in the social utility and social value and trust with important stakeholders. But through this, over the first couple of years of a discovery phase, we'll be developing a best set of biomarkers, either singular or combination, to take forward into a clinical trial. And we'll hear more about that from Vanessa tomorrow. But for this audience today, I want to highlight tier 3 biomarkers. So we will be including samples that will be stored and curated with their metadata, that we would welcome new partnerships, new collaborations from industry or academic sectors to explore, to try and help widen the discovery of biomarkers. So whether this is proteomics or RNA transcriptomics, metabolomics, any other omics or other devices, please come forward, please come and approach DPUK to talk to us about the readout study and whether the tier 3 group in particular may be of special interest to you. Coming forward to the afternoon, we'll see we're really in the process of a revolution. It's not just coming, we're in the process already of a revolution in biomarkers and how they're going to affect medical practice and clinical trials design and trials efficacy. It's an international problem. This reminds me to say that the problems and challenges of dementia are international, and so will the solutions be brought from international collaborations and partnership. And as John has alluded to, our speakers, I think, reflect this international approach. We'll hear later from Cornelia van Duijn on bringing an epidemiological combined with genetics and multiple sort of omics approaches to understand the role of biomarkers in different contexts and different populations. Before that, Nick Ashton. I hope I've not seen Nick today. Everyone else I've seen. Thank you. Very pleased to see you. Welcome. A great relief. So Nick Ashton, currently in Sweden, but about to move to the US, a real world leader in fluidic biomarkers for Alzheimer's disease. I'll speak following Do-Geun Kim from the Korean Brain Institute, and I'm very pleased we have a delegation from the Korean Brain Institute. We'll be talking around the role of inflammation and inflammation biomarkers. But before him, it's a great pleasure to welcome Charlotte Teunissen, sitting somewhere. There you are. Yes, thank you. I was searching to come and really talk under the title of the Biomarker Revolution. Now, Charlotte is a major contributor to the international effort, extremely collaborative. This is pulled from her institutional website of her collaborators across the globe. And through a really collaborative approach to the Alzheimer's Association, the Biomarker Standardization Consortium, through the CORAL Consortium, to developing biomarkers and to training through the Marie Curie Myriad group. So training and capacity development also very important, and that's another feature that DPUK is keen to promote and sponsor. So we should then welcome, please, if you would, Charlotte, to come and talk to us about the biomarker revolution. Welcome.
I'm surprised by the coverage myself, but the worldwide coverage. But yeah, it's actually true. I like to collaborate, so this is also an open invitation. So here are my disclosures. So we have a lot of research support as a group, but also a lot of collaboration contracts. I think also it's very, yeah, it comes without saying that if you want to bring a technology to patient care, be it diagnostic tools or treatments, we need the companies, we need the industry. So sometimes there is some kind of pushback and people are scared of that, at least in the Netherlands, but they don't understand that it cannot come and reach the market and reach the patients without such collaboration. And of course, you have to pay attention to your own will and in research integrity. And I'm also editor of Alzheimer's Research and Therapy. So it has been alluded already, but we need several biomarkers for different purposes along the disease trajectory. So when a patient is not yet diagnosed, we can think of risk biomarkers like the ApoE gene. But when it comes to diagnostics, we need diagnostic biomarkers. But after that, we need probably different biomarkers that we can use to define the prognosis for an individual. Or maybe it's the same biomarker, but then it would be a magic biomarker that it can do everything we want. And we need also monitoring pharmacodynamic biomarkers. So once we start a treatment, we want to know whether it's effective. But also during the course, the natural course of a patient, we want to know where they are on their trajectory. This can also define if you want to apply a different treatment. And we need safety biomarkers once treatments are started. For example, the ARIA monitoring, monitoring of ARIA during anti-amyloid treatments, especially by the antibodies. It's also happening at TREM2 treatments. So different biomarkers are needed along the different stages of the disease. How do we develop fluid biomarkers? There are different stages, and this roadmap gives us also a guidance in that development to approach it also very systematically in order to accelerate this process. But usually we start in phase 1 with the identification of a novel biomarker, which can be hypothesis-based, think of amyloid and tau, but it can also omics-based. And once we have identified our target, we develop clinical assays or research assays that we start to validate in smaller cohorts. After successful completion of that phase, we can move on to the next stage, where we do more extensive analysis in longitudinal studies and more diverse patient populations. Next, we can continue with prospective studies. So now we're really getting close to the clinical practice. And we want to study the real-world performance. And I'll show some data for that for the plasma p-tau. And finally, we want to arrive at the implementation. So then we get into a different realm of guideline development and getting refunding, certification. So all kinds of these questions are then at stake, but they are needed to address to reach the final implementation. And for Alzheimer's disease, we want to measure multiple biomarkers because there are multiple processes. So here's also in the slide, we indicate which biomarkers we were able to measure already in CSF. And so they were already available and they're used in clinical practice, at least quite widely in Amsterdam, but also in different other countries. well, Amsterdam is not a country, but sometimes maybe we think that, but other cities within the Netherlands, but also in other countries. And it helps the diagnosis to establish a biological diagnosis of Alzheimer's disease. And we could measure A-beta and p-tau already for a long time, and it's been very helpful. We can also measure neurofilament light in GFAP, but nowadays we can also measure these biomarkers in blood, be it at lower concentrations, so we need more advanced technologies for that. But nowadays, and this has also brought us at the point that where we are, there are such sensitive technologies, and that has helped and revolutionized the field. But of course, we will also want to map the full complexity and thereby also develop biomarkers for those other processes, so inflammation, synaptic dysfunction, and so on. So here's just one example, and I rely on Nick Ashton that he will show also other examples, but more or less, every study shows the same, and that's the beauty of it. It's a very robust result. So here the plasma p-tau marker, in this case p-tau 181, has been measured in study groups, clinical groups, so defined by clinical definition. And what stands out is that the plasma p-tau is elevated specifically in AD. So compared not only to controls, but also compared to other diseases where tau pathology plays a role. So that's very unique about this plasma p-tau biomarkers. And it's also elevated already in MCI stage. So it's also an early biomarker. And that's shown here, but it's also in other studies. Maybe not so clearly here, but in other studies it is where they're divided by amyloid pathology. So now I come to the point to the amyloid pathology. So just as a summary, because this biomarker does almost everything that we want, it has a very good relation with PET, with very high sensitivities and specificities. So with amyloid PET, but also with tau PET. It also has a relation with cognitive change, so and prognostic value with cognitive change over time. And it has a relation with atrophy. So on all different aspects or different outcomes, so be it clinical, PET imaging, MRI imaging, we see a relation of this plasma p-tau. What's very nice about it is also that the change is not just 10% or 20%, because 10 or 20% is often also variation of immunoassays. But here is a difference of 4 to 7-fold. So it means that we can also allow some variation, some noise within the testing, and still have a good result. So can we use plasma p-tau 217 or 181 also for monitoring of treatment effects? Well, this is a result of one of the trials. So this is lecanemab. I don't know if it's approved in the UK. I can imagine UK is quicker than the EMA, but at least it's approved already by the FDA. And yeah, here at the left side, you see the results, the trial results with the primary outcome on cognitive decline. And the CDR sum of boxes, the yellow line, the treated patients, they have a slower decline, is 30% slower than those who are on placebo. So good effect. If you look at the amyloid burden, you see a really good reduction over the 18 months in the lecanemab treated patients compared to controls. So far the usual biomarkers. Now the fluid biomarkers. So at the left side, CSF beta 42. And beta 42 is normally decreased in 80 patients. And those who are treated with lecanemab, the green line, they have an increase in amyloid beta 42. So that's what we hope for. And if you look at the plasma p-tau 181 marker, there we see a decline because normally p-tau is elevated. So when they are under treatment, anti-amyloid treatment, so not tau but amyloid, we see a decrease in the levels of plasma p-tau. So it looks very promising. But there are still open questions, of course, because we want to know whether this early decline, a decrease in plasma p-tau, whether that relates to treatment outcome. Can you use that early decrease as a surrogate biomarker for cognitive decline. That's what we want to know. Or do you also see it in other trials? Now, the answer to that is yes. But there are still unresolved questions. So this revolution and very quick development in plasma biomarkers, which runs in parallel to the treatment development, has led to the revision of the JAK criteria. So here's an overview of the JAK criteria 2018. And here were mainly CSF and PET biomarkers at the core of the criteria. But these have now been revised, and the criteria are now in press with Alzheimer's and dementia. And you see that plasma has its separate column, it's in it. And we divided the biomarkers in core 1 and core 2 biomarkers. And the core 1 reflecting amyloid deposition, plasma p-tau 217, and we think that's probably the best discriminating biomarker. There are still tests of 181 that are equally good, so I don't want to say that it's always the case, but often p-tau 217 is the most sensitive biomarker. But they are there, and for analysis, we also recommend to use hybrid ratios. I think that's relevant, especially in CSF, because that has been shown over and over that if you use only a beta 42, it's not as sensitive for discrimination of amyloid PET positivity as a gold standard than if you use a ratio like beta 181 over a beta 42. And here are the core 2 biomarkers. And they probably have a role in prognosis and also for monitoring. And there are other p-tau isoforms. Now, in this way, it's 205. I think it goes so quick that this is almost outdated already, but there are many more numbers of p-tau isoforms. So at different positions of the tau protein where the phosphorylation takes place. Probably the C-terminal fragments are related to later stages of the AD pathology, but at least we see that it's highly populated. And we also want to have biomarkers for the different and the other mechanisms that are involved in Alzheimer's disease. So the N for neuronal dysfunction, that was already in ATN criteria, but now we expand it with the inflammation marker, GFAP, is an astrocyte biomarker, but probably also in reactivity in response or part of an inflammatory reaction. Vascular biomarkers, NFL, strictly speaking, is not a vascular biomarker, but it's responsive to vascular damage in Alzheimer's disease. And also alpha-synuclein biomarkers. The alpha-synuclein seeding assay has shown a very good sensitivity and specificity to detect alpha-synuclein pathology. So the table is expanded. And for the staging, we think now that probably the p-tau isoforms are very good and changed already, or very good for diagnostic purposes, because they are changed already in early stages of Alzheimer's disease. And that for the later stages, we probably need other biomarkers, such as the C-terminal tau fragments, but also GFAP and inflammatory biomarkers. That has to be worked on. Staging, we defined biological staging and clinical staging, and they can run in parallel, but probably not completely. And by this graph, we depicted what could be the influence of resilience, for example, and also of having co-pathologies on the staging. So if you have more N, so more neurodegeneration, it could be that moves the bar for the clinical symptoms to the left. So that it increases your vulnerability and that we see clinical symptoms earlier than normally expected if you would have just a pure amyloid and tau pathology. I don't know if that exists, but this is the current thinking and a model that needs to be validated now. So now I will discuss our efforts. some of studies that we do for getting at implementation in clinical practice. So that's the fourth phase of the roadmap. And here's a study that we performed during a year where we collected all our memory or plasma from all our memory clinic patients in the Alzheimer's Center Amsterdam. So no selection, because many of the retrospective studies, they are in very selected cohorts where at least amyloid PET is available. But here we included everyone. So you can also appreciate it from diversity of different diagnoses in this cohort. And we measured plasma peta-217 with the ALSPAT assay and Simoa NFL assay. And what was expected is that the highest levels of peta-217 at the left graph are found in the AD dementia cohort. And the highest levels of neurofilament light would be found in FTD. That's also known, and so that's what was expected. Also in the dementia other, we observed high levels of NFL. And then we made combinations of the two biomarkers, so based on the cut points that we defined, and looked at the frequency of those combinations, because we think that using only those, we hypothesize that only these two biomarkers would help us already with many of our clinical questions. If you look at the right, you see the RC curve for amyloid positivity, SCD amyloid negative versus 80, or A beta negative versus A beta plus. And we see a kind of weird area under the curve for the plasma p-tau217, so it's not perfect. At the left side, you see the effect of the different combinations and how that was distributed among the different diagnoses. So if you look a little bit through your eyes and you look at the light blue color, so the combination of low p-tau and low NFL, that's observed mostly in the control. So SCD negative, amyloid negative, also a bit in amyloid positive. And in the lower bar, you see the suspected psychiatry patients. So they have low NFL, low peta-217. If you look at the reddish colors, there we have the high p-tau groups in combination with either low NFL, so the light or orange color, and the high NFLs in the red color. And you can appreciate that those who are amyloid positive, the MCI patients, they have this combination, and also in the AD dementia, but not all of them. And we defined the AD dementia based on CSF. So it's not a perfect marker, and that's what you can also see in the RC curve. But this is the real world experience that we had, and it's probably due to the more mixed population in this study. It could also be that the ALSPAT assay is not about perfect. When we did in-house comparisons with the Janssen assay and Fujirebio, they were performing slightly better. But this slightly better, I think, doesn't explain this effect. So probably it's real world. Another study that we do is the Cantate project, where we really start with prospective studies in different settings in clinical practice in the healthcare system in the Netherlands. So we want to apply the blood test with the GP, memory clinics, so we call more peripheral memory clinics, or in specialty memory clinics, the tertiary memory clinics. This last one is Amsterdam UMC. And we apply this biomarker panel because we think that the combination of biomarkers is better and gives us more information, even if it does not improve the diagnostic performance, but it does in our models so far. But then still we include GFAP for its prognostic potential, because that's what we observed in our studies that it has. So that's going on as ongoing research. We think it has a huge impact, and you can also imagine it's very logical. So if you have a blood test instead of a CSF biomarker test, it increases the accessibility to all kinds of populations, underrepresented groups. but also it will reduce the overload of the secondary and the tertiary care because otherwise we need a capacity for CSF lumbar punctures or a PET scan, and that's if you have a really massive treatment or good treatments that's available to many people, then we're not able to keep up with that. And this is one of the results of the Cantate project, where we thought of how we should visualize the results and report the results to all kinds of clinicians that will start working with it, and not only the academic ones. And we came up with these two models. So at the left, we have a probability score of the patient that we can calculate. And in this graph, you can see, you can relate the results of this calculation to the overall population. So in this example, we have a p-tau level of 15, GFAP 54, and NFL is Z-corrected, age-corrected Z-score, 1.31. So A-beta dropped out of our models, and that's probably due to the small decreases that you see in plasma A-beta. So we use all these biomarkers and in our model, and the result of this patient is represented by the black line. So you can see that this result fits well to the population that's normally amyloid negative. And we also indicated with a color, likelihood A+, it's low, and it's a green square. At the right, we use upset plots. So you can read it as follows. We give a black dot, whether they are above or below their cutoff, so whether they are abnormal or normal. So if they are all normal, that's at the left side, the far left bar, they're all normal, normal GFAP, normal NFL, and normal p-tau 181. And this is a result that we obtain for a large number in our population that we tested, and it fits well with 88% of who ultimately are normal controls. If you would have a result of all abnormal, that's at the far right, then you fall in that group, and then 83% of this group is normally amyloid positive in our study cohorts. So this gives a clue to the doctors how to interpret the results, and this is what we are prospectively testing, and now already in 230 patients in Amsterdam and Rotterdam. They are using the results, and so far they're happy with it, but we're also systematically evaluating that. Here's another example. Here the results at the far right. So the black line in the probability plot, you see it's at the far right. So the likelihood is high to be amyloid positive. So for this question, and you see also this square of the frequency. We also ask ourselves whether people want to get tested, because that's what's also in public opinion, or some people like to write in the newspaper that we should not test people. So we ask that systematically in focus group and interviews and questionnaires with Alzheimer Nederland. So that's the Foundation for Alzheimer Research Alzheimer in the Netherlands. So we had 107 survey participants, and here's a result for the question. A blood test indicates that you do not have Alzheimer's disease. Would you trust the results in case you have mild symptoms or strong symptoms? So if they have a negative result, but still mild symptoms, they would not trust the results in 84% of the cases. Or they would trust the results in 84% of the cases, whereas if they have strong symptoms, that's a subjective way of expressing it, and I realize that, but then they would not trust the results so much because yet they have those symptoms. So that's an interesting observation. But we also ask the question, would you get screened for Alzheimer's disease, so without symptoms? in the future. And these are caregivers, so they know what Alzheimer is. And if there is no treatment availability, then already 57% would get tested. I think that's already a quite substantial number. But once there is a treatment, 99% would get tested. And that's logical, because then you can do something, hopefully. We also asked doctors in a workshop of the European Academy of Neurology to think of the opportunities and challenges for implementation in primary care, because that's also a lot of discussion whether you should test in primary care, yes or no. So we asked them to think about that. And now here you see the opportunities: non-invasive, cost-effective. They can increase the communication with the family, but also some challenges. And I see them also as opportunities, because those are challenges that we can address. So need for infrastructure, sample handling, guidelines, we can define that. But it's also a call to action that we have to work to that. And another one is potential increase in referrals for further testing. So then you get more diagnostics. Of course, that's not what we want, or we don't want to have too many diagnostics, but yeah, that could also be a consequence. So that's maybe more negative than if it leads to more cost. Funny enough, last week we had a workshop. We asked the same questions to nurses and case managers, and they came up with the same things. So I think these are more universal challenges and opportunities that we have to address. So in my last part, I want to dedicate a few results and words of how to deal with the new treatments. So not only the inclusion biomarkers, that the plasma blood test will help us enormous, but I think there is still need for other biomarkers, like monitoring biomarkers or ARIA replacement, ARIA monitoring and replacement biomarkers. So, and how to accelerate the development of such biomarkers. So first we did a study into how much are biomarkers being used as endpoint markers in clinical trials. And we divided it upon the different phases, so phase 1, 2, and 3, and 4. And I think it's a very clear result that especially in phase 2, you see this green line at the far right corner, that biomarkers are used more in phase 2 trials than in phase 1 or in phase 3 currently. And we ask other questions. How important are they as primary outcome, as in circle C and the pie graph? And as primary endpoints, that's not so often happening, or primary and non-primary, but the largest percentage again in the phase 2 trials. So, but still a large proportion is not using the biomarkers. And I think now that we have a biological diagnosis, we also have to act upon that and use the biomarkers as endpoints in our trials. And also because the clinical scales are not perfect and quite noisy, so I think that the biomarkers can give us a more reliable answer. But this was the situation up to now. What kinds of treatments are we talking about? Now, the biomarkers are used as endpoint mostly in treatments targeting amyloid and tau, and also inflammation. That's what you see here, also as target engagement markers. But there are all kinds of other drugs and targets in trials where biomarkers are not yet used. Even not as a target engagement biomarker. And I think that's a missed opportunity probably. And we can have better trials if we use the biomarkers. So how to develop these? And in the past we used mass spectrometry proteomics, we still do, but we also want to use tests that we can use and commute widely. So that is with the technology that's available in every clinical routine laboratory. And that's usually immunoassays. So that's where we want to end up. But we had a lot of failures with our CSF mass spectrometry results if we wanted to translate it to immunoassay. So we thought of different causes for that. And one of them could be that the technology of mass spectrometry is so different than immunoassays that this causes a disruption of the process. But it can also be due to power. But nowadays, luckily, there are other methods, there are proteomics methods in which you can use antibodies already, and that's by the O-link technology, proximity extension arrays. And yeah, that's again an immunoassay using two antibodies, so highly specific. And you know that antibody pairs are already available if you have a marker. And that would enable speedier development of immunoassays. That was our idea. But still, we need to feed in those arrays and with novel candidates. And I think there the mass spectrometry unbiased proteomics can still be a very good source. So here's an example of a study that we performed to identify novel biomarkers for different dementias. So the cohort size, much larger than what we ever could do in mass spectrometry analysis. It's CSF, so cerebral spinal fluid. We had 200 controls almost, more than 280, 204 FTD patients, really a substantial cohort, and DLB patients also 122. And here's one of the results in our effort to develop biomarkers for Lewy body dementia. And one of the markers, well, there are not so many markers that were different. In the comparison 80 versus control, we had many more, we had hundreds. But here we had almost 100. But one that stood out was DDC. And DDC is a part of the dopamine synthesis pathway. It's the last step of dopamine synthesis pathway. So it's also conceivable that it's related to Lewy body dementia. We also observed several other biomarkers that were related to demyelination, intra and extracellular remodeling, and also immunity. So we next move on to develop a panel, because with O-link technology you can design your own panel of maximum 21 markers. So we calculated how many we need, and if we need 21 or less. And we found out that we need only 7 biomarkers for the optimal performance of DDC versus control. So if we add more biomarkers, it would not have any added value. And DDC is already a very powerful marker, because here you see the area under the curve for DDC. If you compare it DLB versus controls or versus AD. AD is in the pink line, and the comparison with controls is in the purple line. But we had this 7 biomarker panel. You also see the confidence intervals of the reiterations and the calculations with a very high area under the curve. So if we add 6 other biomarkers on top of DDC, we still have a better performance of the test. And we observed a similar reflection of the different pathways. We validated this 7 biomarker panel, so we developed a customized panel of those 7 biomarkers, and then we next tested that on different cohorts. In cohort 1, it was a very nice validation, and cohort 2, somewhat lower area under the curve, which could be related to the clinical diagnosis of the DLB in that specific cohort. We also validated in a cohort where we had autopsy confirmation, and we see a very high area under the curve. We then developed an ELA assay, so that's also an immunoassay platform, microfluidics, and also a Simoa assay. And here are the results of the ELA platform. And here we see again confirmation DLB of DDC levels are highest in DLB, and compared to control and AD, a very high area under the curve. And we see a very strong correlation for this cohort between our ELA assay and the original O-link discovery results. So this this is proof of our idea that using this O-link platform helps us to accelerate or at least get a successful result with a single immunoassay that we can develop. Lastly, I want to share these results. So we not only developed a panel for DLB, but also for AD. Again, also in a small panel on the O-link platform. At the far right, you see the result of the custom panel. And again, a very high area under the curve, so it works. And I think this platform can also help you with defining what are the mechanisms affected by specific treatments. So in this case, it's a treatment also against a PDE9 inhibitor, but in the DLB population. I'm not going into all the different biomarkers. It's a lot of detail, but it's a very small cohort. I think that's important to see. And in the left graph, you see the changes over time. And those who are amyloid negative, so DLB amyloid negative, so pure DLB. And in the right volcano plot, in the B graph, you see what's happening in the amyloid positive individuals. And you see that there is more changes in the amyloid positive individuals. So it's depending on your amyloid positivity, what's happening with all kinds of pathways. And very important, the pathways that were affected, we could also track them because there were multiple markers related to those pathways. And if you look at the left graph, you see the changes over time. Those who are amyloid negative, we see some change, but in the now pentotenate and coenzyme A biosynthesis. But in those who are amyloid positive, you see a more strong increase. And in a GABAergic system, we see an effect on those who are amyloid negative, but not in those who are positive. And those results are significant, even in these very small cohorts. So it looks very promising. And I think we can still do, this is typical phase 2 size or phase 1 size of a cohort. It can give you a lot of information, and you can also consider developing panels based on this results, or those kinds of results. So now I come to my conclusions, and I added some illustrations so that you recall what I explained to you. I think it goes without saying, plasma biomarkers for AD develop at a really high speed, and it's very timely, the advent of the disease-modifying treatments. And we need a panel of biomarkers to capture the full complexity of Alzheimer's disease, but also of other dementias, because patients do not come at the doctor only for knowing whether they have AD. The doctor also has a question, okay, if it's not AD, maybe it's suspected FTD, and neurofilament light is a very good example for that. And we need education and interpretation tools. We developed our upset plots and probability graphs. That could be a way to represent multiple biomarkers. And yeah, maybe also for multimodal biomarkers, we have to think further. Also how to include digital biomarkers, for example, and those results. And I think we need to exploit the value of biomarkers more in clinical trials. And that's it. A nice hello from my neurochemistry lab in Amsterdam. Thank you for your attention.
Thank you. Thank you very much. Very exciting talk. I'm relatively remote to the biomarker field, I would say, but I think there's some evidence about variability of biomarkers depending on the time of day. For example, sleep has a big impact on biomarker concentrations, especially in the plasma. In any of your essays, do you actually control for day of time of sampling, basically?
Yeah, for CSF, that has been shown in the past, but it was not a very consistent picture. So there was variation within every patient, but a different pattern within every patient. For plasma biomarkers, indeed, we have to be more cautious because there are more direct influences also of diet. There are not many studies yet performed, and I'm not so sure whether it really has a very strong effect. Even if you look at night and day, yeah, there can be effect, but no one is undergoing a venipuncture during the night, usually. but because Alzheimer's is not a acute disease, so it's quite tight time window, you could say, between 8 and maybe 3 or 4 in the afternoon. So even if there is an effect, maybe it's not that big. But the effect of fasting, I'm not yet convinced that it has a major effect on these biomarkers.
You do not have the
We
information on that.
don't, because we never asked people in our biobank to but it needs to be studied. That's a good point.
Good afternoon, everyone. I'm very honored to speak in the front of all these very world-renowned researchers here. A little bit, it's not fair to have it. It's not fair to have a presentation after the giants of the research. And I'm a little humbled by some of the people who are in front of me, but I'm just trying to give some of the information that what we're trying to do. Especially, I'm from South Korea, and we are at the verge of moving from the basic research into the clinical studies. And I'm going to give some of the showcase that we're having in our institute. So the outline for this talk is I'm going to introduce, because a lot of people may not be familiarized with Korean Brain Research Institute, so I'm going to introduce our institute briefly. I'm going to introduce some of the collaboration in between Gyeongbuk National University, which is quite close to our institute, and some of the research data which were done by the omics study, including the AD and PD, and what we're trying to do in the future. So let me briefly introduce KBRI. Now, I already came here to collaborate with Professor John Gallacher last year, but people from the DPUK may about our institute, but a lot of people here may not know about it. So let me just briefly introduce KBRI. So Korean Brain Research Institute was just established in 2013 and is located in the Daegu, which is a southern part of the South Korea. Now we are working in the both in basic research and applied research, and at the same time we are trying to move into the implemented research, which is going to the step 3, as we can see here. We have a research groups, which is about 8 research group, which is most likely focused on the basic research, including the neurovascular unit, circuit, sensory motor system, developmental disorder, and including them also dementia research group. Also, there are a lot of collaboration that happens in between our institute and other groups as well. And also, we are working with other international groups, including the DPUK. So also we are trying to move on to the database research as the DPUK does. We are trying to recruit a lot of the patients and to get some clinical samples and getting the data for the clinical research and some of the a lot of imaging data and etc. And we are trying to merging them and try to get the information out of it, which might be helpful to get the medicines and which might be helpful to deliver in the industry. That might be more like advanced neuroinformatics system for a global brain research collaboration. I've been just talking about what we are doing in South Korea, but at the same time, I wanted to give some of the focus on the collaboration that we have in between our institute and the Gyeongbuk National University. This has it has been developed based on some of the research collaboration that was given by the Korean government. And the Korean government wanted to have some of the information and clinical data-based study. And we had because Gyeongbuk National University, this is National University, which is located in the Daegu, and it's very close to each other. It was very good opportunities for us to collect the samples from a lot of neurodegenerative disease. So one of the purpose for objective of this research was actually having the longitudinal data set, because we know that the Alzheimer's disease start from the normal patients, and they're having the MCI, and they move on to the AD, but people do not know what happens in between the MCI and the AD. So we wanted to have a better biomarkers, what happens in between those period that moves from the MCI into the AD, MCI to AD period. So it has started, it has been a long time for us to collect the data set. So we are planning to have it for a very long time, like 10 or 20 years from now on. So we have been collecting data set from those patients. And we have gone up to 3 years for now, but we have collected about 300 samples from patients, including AD, PD, and MCI, and including we also collected samples for ALS as well. So as you can see here, we have quite a bit amount of samples for the AD, and also we have some follow-up studies. So one of the importance of these studies is that we are having follow-ups. So some of the patients who were visiting the Cambridge National Universities, and they're coming back after years, and we naturally collect the data from these patients who comes back later on. So we are generally, as time goes by, we might be able to get the follow-up studies, which might be very helpful for us to get what happens in the long run. That some patients who are having the MCI, some of the patients may move on to the AD, and we might know what will happen in those periods because we're collecting the blood samples, CSF, and at the same time CBC circum data. And sometimes we might be able to get some of the MRI data set for the follow-ups. So those data might be very important for us to interpret what happens in between the MCI and AD. So for those samples, in order to increase the validity of the samples and integrity of the samples, we're also checking all the ATN data for the CMOA, using the CMOA. So we are validating whether they're all having the AD using these biomarkers. And at the same time, as I've been showing in here, we're also taking the amyloid beta PET as well to differentiate whether these patients are also having the AD as well. Also, because we were having samples from them, so we were collecting the blood samples, the amount of samples have reached up to 270 in the 2022. So we have made up that we're going to give some the proteomic analysis for these samples as well. So at first, we were starting from the 80 samples. So we were having healthy control, which was around 60 samples, and then having MCI, AD, about 50 patients. Now we were collecting plasma samples, and we were doing sample preparation, and we were depleting the BSA, and we were doing all this preparation in order to get the protein samples. Now we were doing the Alzheimer's analysis, and also we were doing some of the algorithm-based alignment in order to give better analysis on these samples. So I forgot to tell you, but these were done by our collaborator in the Mugyang Chan, also Ugyang Yu in the Digist, which is quite close to our institution. So we were using the match between runs algorithm in a Mexiquant, which was used for align those samples, which were having different values and based on the runs of the Alzheimer's analysis. So we were using this algorithm, which were done by Woo-Kyung Yu. After using this algorithm, we were able to get the more aligned data set, which were very helpful in order to get the better analysis data out of the Alzheimer's analysis. I haven't put all the data sets in here, the names of genes, which might be very helpful for you to understand what I'm trying to say, but just forgive me because they were under patent, they were before the patent and stuff like this, so because of it, we haven't put the data sets in here. But after the analysis, what we have found from these histology scale proteomic studies was a lot of genes that were involved in this out of these analysis were actually the genes that were involved in the coagulation pathway, and a lot of them were involved in complement pathway, which might be involved in the inflammatory pathways in from these samples. And then one of the interesting thing was that, as you can see here in the normal control and the MCI, you barely see the differences of these gene sets in here, and especially these genes that were changed in here or failed to form the pathway, which were having some disease pathway. So we were trying to get to some of the analysis in the long run. There might be some of the changes what might happen in between the MCI samples toward the AD-like pathway formation that we're trying to see what happens until later on, after 10 years or 20 years later on. Based on these, as I said just briefly, one thing that was a little bit surprising to us was the pathways that were classified using these the Alzheimer's analysis. A lot of them were actually that were involved in the coagulation that are known to be very important in the atherosclerosis, other cardiovascular system. Also, there are genes that are involved in the the amyloidosis and some of the vascular defect disease like patterns in here as well. Also, when we were putting this into the CAC pathway and the other pathway analysis, now what we were what we could see was a complement in the coagulation pathways, and some of them were they were involved in the cholesterol metabolism pathway. And also, a lot of the genes that were activated were classified into fibrin clot formation, indicating that a lot of genes that we could find from these pathway analysis is actually involved in the coagulation pathway and abnormality. So what happens for the coagulation pathway abnormality? So a lot of studies, although this is not major pathway, people are focusing on the Alzheimer's disease, but there are several studies which are showing the importance of a defect in coagulation and its impact on the Alzheimer's disease progression. And there are some of the studies that were done by us. Also, a lot of studies, some of the studies that were given by Katrina Kalasoglu, which are who is in the UCSF, and then she was showing that the importance of fibrinogen, and which is a coagulation factors in the induction of Alzheimer's disease. So also some of the studies. So this is one of the importance of doing the research using the animal models as well. Some of the studies that were done by our groups, and we could see that in the PD mouse brain, with PD mouse model brain, we could see the accumulation of fibrinogen in the brain parenchyma in the 13 months after birth. Also, in the 5XFA mouse brain, we could see a lot of accumulation of fibrinogen into the brain, meaning that these defects that we could see from our study in the clinical samples might be tightly linked to the pathway, disease pathway in the animal models as well, which might need further studies in the clinical samples as well. And now one of the surprising thing for us was actually the difference from the data set that we could see from the that was already published. And a lot of data set were having some of the gene set that were not similar to ours. However, we have to know, we have to do further studies whether our research data are having some of the other gene set that we haven't found from our studies. But we might need some of the inputs and help from the other people from this audiences here as well, who are experts in this field. So our plans are, samplers are going to be assayed further, and as already the previous speakers already talked about, was the O-link assay, which is very helpful. Especially a lot of Alzheimer's data they're having some of the hard times for analyzing data based on the quantification, because the methodology they're using is based on the fragmentation of the peptides. So we are trying to move on to the O-link analysis to have a better predictability and the precision. Also, we're trying to do the validation using cellular models and animal models, which is a very strong point from our institution, and which might be one of the collaboration ways that we can do with the UK-based research institution. Also, we're trying to narrow down the biomarkers because there are a lot of genes that we have found from our studies. We have to narrow down the number of genes that we have found up to 3 or 4 genes, which might be used for the biomarkers for the AD, which can be used for prediction of the AD progression in the long run. So another study that we did was using analysis for the Parkinson's disease. So this is a little bit flip-flop. However, the reason why I've been trying to move on to this was because we were trying to do the in-depth study. However, the in-depth study is still ongoing using the animal model-based studies and other validation studies. And we had another studies that were going alongside, which was the Parkinson's disease. So these are the heat map that we have analyzed from the open source data bank, which were given by Parkinson's Disease Association from the Michael J. Fox. And a lot of genes that were found from these data sets are actually heavily related to neuroinflammation. And a lot of some of the studies that we have done using the alpha-synuclein, which is a core proteins which has important roles in the progression of Parkinson's disease. And when we were treating the alpha-synuclein on this brain endothelial cell, we could see a lot of increase of pro-inflammatory cytokines in the brain endothelial cell. So that we had samples for the Parkinson's disease, and we just wondered if these kind of pro-inflammatory changes are also observed in the Parkinson's disease blood as well. So we were applying for, we have done all the analysis for the LCMS analysis for Parkinson's disease patient blood as well, but we're doing the further analysis as well. Well, in the same time period, we have focused on the see if these types of neuroinflammatory responses are also observed in the blood as well. So we use O-link analysis from the samples of the PD patients. And we have used this technology in order to narrow down the pro-inflammatory cytokines from the panels from the blood samples as well. And we followed these the pathways in order to analyze the the blood samples and get the types of pro-inflammatory cytokines that are increased or changed in the Parkinson's disease patients as well. So these are the heat maps from the Parkinson's disease patients' blood samples. And these are the samples. I'm sorry for this, but I removed the names of these genes. However, out of 96 samples that 96 pro-inflammatory cytokines, we could see 11 different cytokines that were changed up in the disease progression, indicating that pro-inflammatory cytokines are indeed changed in the blood samples from the PD patients as well. And according to study from the meta-analysis on the inflammatory biomarkers in the Parkinson's disease, compared to the AD, we do not have a lot of studies in the biomarker studies in the Parkinson's disease. However, there are some reports showing the strong links in between the cytokine and the development of Parkinson's disease. And out of the genes, we have found at least 3 different cytokines that are involved in the autoimmune disease phenotypes. And we could see 3 different genes were not highly, however, at least 2-fold differences compared to the its control groups, indicating the potential possibilities for its important roles of these pro-inflammatory cytokines in the development of Parkinson's disease. And then when we were combining these pro-inflammatory cytokines, the accuracy of the prediction of the Parkinson's disease was increasing up to 90%, indicating that although we are using alpha-synuclein as biomarkers for the Parkinson's disease, however, we might be able to use this methodology as the complementary method in order to predict the progression of Parkinson's disease in the long run. We have to validate using these biomarkers using the cellular and animal models in the long run, and also we have to narrow down the biomarkers and have to have the finding reach to the point where we have to find the optimal combination of biomarkers that can predict the PD with higher accuracy and precision. So what we have to do in the future, and then what we're trying to do is we're trying to make a consortium with the hospitals. So we are just working with Gyeongbuk National University. However, we are trying to make a consortium with hospitals in South Korea and increase the number of the cohorts that we can have the better clinical samples and the data and a lot of clinical data sets from these patients in a longitudinal manner, as the DPUK does. We're trying to do the cross-validation from the different cohorts, and we're trying to apply advanced analytical technologies especially omic studies like the O-link assay to analyze the sample with a better precision. And also, we have to do the validation of biomarkers using animal and cell-based assay. And apparently, we're just a beginning phase, and we have to learn a lot of technologies and ways that we can validate these biomarkers. Now, we may need a lot of help from the colleagues and the institutions which are affiliated in the DPUK, which might be very important for us to be successful in the long run. So with this, I would like thanks to the lab members in our lab. And at the same time, a lot of these samples were acquired from the help from our research group. Also, Professor Huan Li from the Kampong National University. And I would like some questions from the audiences.
Thank you for the fascinating presentation. I'm an old age consultant psychiatrist. Sen is my name. Just wanted to ask regarding inflammatory biomarkers in vascular disorders or vascular cognitive disorders. Have you found anything?
From the vascular dementia samples as well? Yeah. So it's one of this very hard, very important question, but at the same time hard to answer question because the judgments to decide which one is the vascular markers are very important question. Whether we have to focus on some tight junction molecules or something which are harming the integrity. But are you asking some of the pro-inflammatory cytokines which are released from the brain endothelial cell, or is there some question?
If you can give any insight into that, that will be great. Doesn't matter.
Yeah.
Whether any.
Yeah. So there could be some chances that it's really hard to answer. However, I'm working on blood-brain barrier, and that's my major topics that I'm very interested in. And I've been talking this with some of the experts in this field, which could be the clinical markers which can detect, which can be used for the vascular defect. However, one of the best methods should be just using the MRI, which at the early stage of the AD, I mean, that could be one of the best markers to use because at this time it's very hard to do. Or, as previously the speaker already explained, some of the integrity might be harmed when we are looking at the NFL, neurofilament light chain, and if they are found from the plasma, that could be one of the ways that we can use as vascular markers. But I mean, I've been talking this with experts from the UK as well, but she also told me that it's very hard, but we're working on it, so it will take time. That's the only answer that I can give for now. Yeah.
Thank you.
Yeah.
Any other questions? If not, I wonder if I could just ask you to comment on the specificity or generalization of this pro-inflammatory cytokine pattern. You touched on PD and AD, other dementias. Do you expect the different diseases to have a unique signature, or is this a generalized pro-inflammatory response to all the neurodegenerative diseases?
That's very hard to answer, but I would say we might do some of the kinetic data, and at the same time some the cross disease analysis for that. But for now, I haven't seen any data that were focused on it. That could be very important topics that we have to work on. But I personally believe that it should be disease specific, but I may need better studies, further studies to answer that question.
But well, just to know what you anticipate is extremely helpful to have your insights, to have you share those. So thank you very much, Do-Geun Kim.
I'm going to be talking about blood biomarkers, surprise, surprise, and I hope that it would be very complementary to what you've just heard. And we're talking a little bit more on the specifics around phospho-tau. And I think everyone in this room at some point is going to be using blood biomarkers, and if you're not used to it or care about it, I hope you take something away that you'll be able to use in the future. So 3 main topics I want to go over is why and how have we settled on phospho-tau 217 in general in the field? And what does phospho-tau 217 mean when it increases? We all know it means Alzheimer's disease, but what does it specifically mean when it increases in the blood? And how do we improve on phospho-tau 217? Is it the answer to every problem we have in dementia? I'm sure from Charlotte's talk we know that the answer is no. So how do we improve on this? Before we need to think about our biological definition. So Alzheimer's disease in the research space and increasingly into the clinical space is no longer based solely on the symptoms, and we look at the underlying biology, and that arose from Alice Alzheimer's observation and up to the current day where we have this ATN criteria, which we summarize amyloid and tau and neurodegeneration by imaging and CSF. And of course now we're looking more progressively in blood. And if you look at this countdown clock of accumulation of familial Alzheimer's disease patients over time, so we're 20 years before their symptom onset, I hope you can appreciate here that the amyloid accumulation in these individuals in the precuneus and other areas of the brain is rapidly increasing decades before symptom onset is occurring. So if you look at this clock countdown, as we hit 0 now, I want you to just observe on the amyloid deposition. There's almost no amyloid deposition after 0, or it's saturated from the imaging. So this tells us that the onset, or what we're trying to measure and clear in our drugs, the majority of this is happening before symptoms has occurred. Here are approved or soon to be approved drugs that clear amyloid, and whether you think this is the right target or not, we need biomarkers and need to ensure that at least we have the right patients that are receiving those drugs. So blood biomarkers, everyone would have heard about it the last few years, maybe sick of them, in fact. But this is a 20-year story. So this is the Daily Express. Whether you believe what the Daily Express says or not, I presume that most of you don't. But you should, because these are 2 studies that I performed on the front of them. Similar headlines, both showing scandals in the government, so some things never change. But 2 blood biomarker breakthrough in Alzheimer's disease. One was in 2014 and one was this year in 2024. So what's the difference? Why has this not been established for many, many years? Well, in 2014, our studies were looking something a bit like this. We're using 2-gel electrophoresis, very low specificity, measuring high abundant proteins, and making inferences about clinical diagnosis of Alzheimer's disease rather than a biological diagnosis. But even then, well, most of the studies could come up with a differential between Alzheimer's disease controls. And this, as the last talk suggested, highlighted the complement system, the inflammation system, and this kind of set this cascade of biobanking and storing blood samples that would have a specific biomarker in the future. And this kind of summarizes that decade of effort, that we have a lot of biomarkers that were suggested in models, not highly replicated, but they were related to Alzheimer's disease in some way, and they were probably telling us something secondary about the disease, whether it was poor kidney function, cardiovascular disease, not specific to the pathology, but someone living with Alzheimer's disease with poorer health. And they have now we know with our core biomarkers, they had a very poor correlation with what we have. So we needed something a bit more specific. And this is where phospho-tau217 and other biomarkers have come into this. So this is really what our makeup looks like in clinical chemistry, particularly in Sweden. This is what we measure in the cerebral spinal fluid, and on the right-hand side is what we can do in blood, mainly academic, much important to point out. But what we're doing in CSF, we're catching up in blood where we can have phospho-tau markers, many of them, amyloid, GFAP, NFL. We're even starting to get an idea of the synaptic pathology in blood as well. So we're rapidly catching up with what we can do in CSF. There are some important gaps, of course. But I want to take you very, very quickly through 6 years of rapid progress in developing a phospho-tau biomarker. These were our first studies, which Charlotte has already presented to you in 2018 to 2020. Large cohort studies focusing on phospho-tau 181. They were proof of principle that this biomarker would work with high specificity, and they were validated against gold standard, so post-mortem tissues. 2021, we started to discover more epitopes in blood, just because of the accessibility. So phospho-tau231, phospho-tau217. We were looking in preclinical studies. We could see that these biomarkers were changing as amyloid was increasing in the blood, was increasing in the brain of people without symptoms. 21, 22, we started to have mass spectrometry methods, so we could have greater target specificity. That's what mass spectrometry gives you. It doesn't give you greater sensitivity, but you know exactly the target you're measuring. And that's a multiplex assay, so you can measure multiple phosphoepitopes. We had longitudinal data, and really importantly, we started to see that phospho-tau 217 and 181 were decreasing after you cleared amyloid in these clinical trials. So it was an explorative endpoint in these trials. So there was a decrease when you cleared amyloid. And coming up to the present day, we have a lot of competition. I'll talk about this in a bit. 33 different phospho-tau assays for blood biomarkers on the market, available commercially or posed to be commercially available in the next few years. We'll go into this in a bit more detail. We started to learn a little bit about some of the negatives about blood biomarkers, kidney dysfunction, other things that will affect biomarkers. But importantly, with the Alzheimer's Association and led by Charlotte, we started to have guidance and appropriate use criteria, how to use these biomarkers and when not to use these biomarkers. And what about today? So in relatively 6 years, we've gone from academic, I would say, assays to fully automated assays on the Lumipulse or the Roche Alexis. And these are workhorse machines that sit in our clinical chemistry labs today. The same tests that you do CRP on, now you can do phospho-tau 217. Now, this is data we haven't published at the moment, but this is how the Lumipulse looks across 5 different centres in 2,000 individuals. We've driven one cutoff in one centre and then tested them in multiple centres using this 2-tier cutoff. As you can see across the centres that the positive predictive value and negative predictive value of this fully automated test, so nobody touches the sample, goes from the doctor's office into the machine with a result within 20 minutes, is extremely high. But it can't be all positive, and it's always good to have self-criticism. So this is my self-criticism slide, and I've reduced it to one animation, just so I'm not too self-critical. There are some things we need to consider about plasma biomarkers. I've listed them here, there are probably more. Kidney dysfunction definitely affects the levels of these biomarkers. That's undeniable, that's proven by many studies. Whether this is clinically significant or not, I still think needs to be proven. Fasting versus a non-fasting state does change the levels of the biomarkers. Again, we need to know whether this is clinically significant. To the question earlier about dynoa variation, we know that there is a slight change over 24 hours. We've done one study where we've measured a blood every hour for 24 hours. Don't ask about the ethics about that, but it was a great study. And these biomarkers change about 10 to 15% over that 24-hour period. That's a problem for some biomarkers like A-beta, but for a biomarker Charlotte described, which changes 4, 5, 6-fold, that's not a problem. And immunoassay design I'll talk now, but this is something I really want to show, which is important for clinicians. So this is a study we've performed in healthy individuals, fabulously described outstandingly healthy, which is British for amyloid negative, I think. So these are 40 individuals, and this is just one of them, that have had their phospho-tau levels taken every single week for 10 weeks. And most of the individuals looked like this, remarkable reproducibility and standability across that time. But we have 1 or 2 individuals that look like this. Again, amyloid negative, but we have a spike, and it comes back down again the week after, and another spike. And this happens to all phospho-tau assays. Didn't happen for NFL or GFAP. Now, this is not sporadic Alzheimer's disease that disappears next week. It's also not technical, because we know that it happens to all phospho-tau assays. So this is something physiological that's happening, that's making this biomarker peak at certain points. So it's kind of led us to say that our recommendation is that if you have a positive test, then maybe this needs to be repeated the next week just to ensure that it stays positive. So these are kind of the things, the nuances that we're learning. What about the other biomarkers? Why am I so insistent on phospho-tau 217? We have multiple things that we can measure in blood. 181, 231, there's 205, and there's phospho-tau 212. So what are the differences between them? And essentially, there are very little differences between them, I would say. But there are subtle differences, and when you are measuring massive populations, those differences become very important. So this is this round-robin study that we've performed with the Alzheimer's Association. We've measured 33 different phospho-tau assays in plasma and in CSF. It's a small study, but it illustrates the point. All the phospho-tau 217 assays are in green here, and this is fold change between Alzheimer's disease and non-Alzheimer's disease. So all these people have symptoms. and this is the fold change in CSF. And as you can appreciate, all the phospho-tau217 assays are right at the top there. They have the largest difference between AD and control. So that gives us a much better test, easier to understand, higher diagnostic accuracy, more importantly, better positive predictive value and negative predictive value. And I would say in general that we have more 217 tests that are better validated and are more robust than other tests. And as Charlotte has pointed out, we have some other epitope biomarkers that also perform just as well, and that goes down to robust assay design. Another good thing about these 217 assays is that they highly correlate with each other. That's really important if we want to develop a certified reference material. So if we want to standardize tests across the world, across labs, we need assays that correlate. And these biomarkers are correlating very, very well. And that gives us good hope of being able to standardize different tests around different labs and calibrating them to one single value, like we do with CSF and like we do with PET, like the Centiloid value. So can we do this at home? Is this a bit controversial? Now, I'm not saying that we're going to get people to read out their own 217 results at home and write it down and send it in. What I'm suggesting is that maybe we could make the blood collection a little bit simpler. We still rely on quite strict protocols for collecting blood. You must have it done from the venipuncture. It must be done by a nurse, spun, frozen, etc., etc. So can we simplify this method? And that is what we've been working on with this DROP-AD protocol, which is essentially a capillary blood draw, which is stored onto a card, and that is stable at room temperature for about 3 months for different biomarkers. Then it can be sent to a lab and processed, and we essentially use the same assays as what we're using for normal blood tests, so we haven't reinvented anything here. And I don't want you to go away from here saying we don't need to collect blood from venipuncture because Nick said you can do it through your finger. Now, that's not what I'm suggesting, but what I am suggesting is it could be a possibility in the future, because this is the kind of results that we're getting. This is the association between a normal blood draw for phospho-tau217 and from the capillary card, and this is the separation between amyloid positive and negative people with a capillary card, and the area under the curve is lower than a normal blood test, but not substantially lower. So this is work in progress, but looking very promising. Through the same blood spot card, you can also measure GFAP and neurofilament, and also get an idea of if you're interested in frontal temporal dementia, if there's a progranulin mutation carrier or not in there. So to the main point here, what does phospho-tau217 mean? We know it's a robust marker. We know that if it's high, you're likely to have Alzheimer's disease pathology. But what does it mean mechanistically? And we need to now understand that, because if there is a change in the biomarker or a decrease after a treatment, we need to understand mechanistically what that means. So is phospho-tau217 a marker of amyloid? That's what most people suggest. And before I say this, I want to say that this is not causative data. This is associations with pathology, amyloid PET. So I'm not done any sort of mechanistic studies to make these conclusions. This is just what we know from our cohort data. So is phospho-tau a marker of amyloid pathology? So what are the arguments for this? Well, phospho-tau increases at very, very low levels of amyloid PET burden. Around about 12 centiloids, you start to see increases of phospho-tau in the blood. Presumably, there's not much tau pathology present there, and this is very, very subtle. This is under the most thresholds of amyloid positivity. So this would be one argument for that phospho-tau reacts to amyloid. In genetic forms of the disease, they increase even earlier. So in Down syndrome and familial Alzheimer's disease, 20 to 25 years before you see amyloid deposition increase phospho-tau increase in the blood when amyloid is depositing in the brain. And from animal models, we know from these APP-PS1 transgenic mice, which do not form neurofibrillary tangles and don't get neuronal dysfunction or death, that phospho-tau increases in the blood and cerebrospinal fluid of these mice. So even when tau is not present, phospho-tau is increasing as a response to amyloid. So is phospho-tau a marker of tau pathology then, or is it just amyloid? This is very, very difficult to prove, and you'll see from my very, very elaborate examples of this, of how this is difficult to prove, because of course, phospho-tau doesn't increase in primary tauopathies. Charlotte has shown that, so PSP and CBD, we know that that doesn't increase. But it's very hard to find cases where of Alzheimer's disease diagnosis where you have no amyloid and tau positivities. These are extremely rare. So it's difficult to deduce if phospho-tau is a tau marker. But if you look at Niemann-Pick's disease, which is a lysosomal disorder, they have neurofibrillary tangles developing in the brain, 3R4 tau, Alzheimer's disease like tau, in the absence of amyloid. So we've looked at these individuals, and we can see quite clearly here there is an increase in phosphorylated tau in the blood. So this kind of gives us evidence that there is an association between phospho-tau and tau deposition in the brain. Now this is my elaborate and reaching claim for phospho-tau being a tau biomarker. If you look in hibernation, like hibernating squirrels, red squirrels, they have increased phospho-tau accumulation and PFH tau in the brain when they hibernate, and when they wake up, it's completely cleared. This is a known fact. There's a whole field looking at hibernation as a mechanistic reasoning for curing Alzheimer's disease. Unfortunately, the red squirrel doesn't have the same sequence as human tau, so our immunoassays didn't work. But we have some brown bears in Sweden that do have exactly the same tau sequence as human taus, and they also form tau pathology when they hibernate, which clears when they wake up in the summer. So some poor PhD students were going around darting bears in the north of Sweden, collecting blood, sneaking into the den in the winter, collecting blood again. It's not far from the truth, but yeah, it was done with the conservationists, of course. And we see very, very high increases of phospho-tau when phospho-tau is depositing in these brown bears as a response to hibernation. No amyloid present. But the most realistic is that phospho-tau reflects both. And this is, again, an association study, not a causative study, by the Biofinder group. And what they looked at was neuropathological deposition of amyloid and tau and how that reflected the blood changes. And essentially what they found is that if tau pathology was restricted to the medial temporal lobe, that phospho-tau and tau did not associate, but there was a good correlation between amyloid and tau. But later stages, as phospho-tau spread across the neocortex, say Braak stage 5 and 6, this is where the association between tau pathology and phospho-tau in the blood started to become very, very strong. So it has this dual mechanism, amyloid association in the early stages of the disease, tau association in the late stage of the disease. Okay, so that's that answered. Great. So is there anything else where phospho-tau 217 increases? Now this could get a bit complicated and a bit nerdy, but I think this is important for us to understand. So there are a few studies to show that if you have motor neuron disease, that phospho-tau increases. Me personally, I don't think that this is CNS phospho-tau that increases. This is peripheral tau that increases. So what I'm showing here is different types of phospho-tau assays in the blood. Here are two 217 assays. One, there is no increase in ALS, and the other one, there's a very, very large increase in ALS. Same with 181, low increase, a very, very large increase. So what's the difference between these? They're measuring the same thing, supposedly, so what's the difference? So we have two types of tau, basically. This is brain-derived tau, and this is peripheral tau. And you see that peripheral tau has this big exon 4 insert. And this is how our immunoassays work. So the capture antibody binds phospho-tau, so this is around 217 or 181. And then it has a detector antibody. Now, some assays are capturing what we call brain-derived tau, but some immunoassays capture the N-terminal part of tau. So if you put this on peripheral tau, this is actually wrong. This one should be here. So some assays have specific brain-derived tau connected to 181, and other assays have N-terminal tau, which will encapsulate the whole of the exon 4 insert. And that's what happens with these assays. They are also capturing peripheral type of tau rather than just CNS tau. So not only do you not need to know what biomarker you need to measure, you need to be, have some good advice about how your biomarker or your assay that you choose is made, basically, and what it really detects. And I'm sure I can give you Charlotte's number, and you can call her and she'll let you know anytime. So that was more of a assay design fault, I would say, with phospho-tau, of why we might see some increases. But we do see some increases in acute neurology. So We've known for many, many years that neurofilament light and total tau in serum are excellent prognostic markers in cardiac arrest. So unconscious high levels of neurofilament light means you're more likely to have a poorer outcome after 6 months. And phospho-tau really increases at 24 hours, but has this almost opposite effect. It's then rapidly cleared from the brain. So this is telling us that if there's a defect in the blood-brain barrier, the phospho-tau will rush into the blood system, but it will be rapidly cleared again. In TBI, we see something quite similar. So phospho-tau increased at day 1 after a big traumatic brain event. It's similar levels that you would see in AD, but after day 1, after day 10, these levels are back down to normal levels. So acute events would increase phospho-tau. And in newborns, very, very high levels of phospho-tau in newborn babies. And the reasons behind this is not known, but it could be for normal neurite growth or some sort of development tool. But as you get into young adults, the levels are normal and they start to creep up with age again. Okay, so finally, is phospho-tau the answer for everything? And the answer, quite simply, is no. I think that phospho-tau is extremely good for one very, very needed purpose. Do you have Alzheimer's disease pathology in the brain or not? Or does this patient sitting in front of me have Alzheimer's disease pathology in the brain or not? And it's extremely good at doing that. And I hope the last few talks have shown that to you. But there are a few things that it's not very good at. And this may come as a surprise because I've shown it and Charlotte's shown it that phospho-tau decreases after amyloid clearance. And these data that we've shown were at the group level. Now this, thanks to Lily and Mark Minton that allows to show me this. This is at the individual level. So a decrease in phospho-tau 217 to 24 weeks and a decrease in amyloid PET signal to 24 weeks. And there's absolutely, or they've got a significant association there, but you can see it's very, very messy. So phospho-tau in the blood is not exactly mirroring the decrease of amyloid in the brain at 24 weeks after these amyloid trials. So we need better outcome markers. Longitudinal change, again, at the group level, we see great associations. But if you look very, very sharply at these very light longitudinal lines that I drew as well, you'll see that the data is quite messy. So phospho-tau explains the most variance in longitudinal change, but it doesn't explain all the variance. As we discussed, phospho-tau doesn't answer primary tauopathies. So it just simply doesn't increase. Unless there's amyloid pathology present in these green dots, then you will get an increase in phospho-tau. Phospho-tau will not be the answer for other drug targets, multiple drug targets that we have looking at Alzheimer's disease. And potentially neurofilament could be the answer here, but we need some biomarkers for adverse events. Can we predict those that would likely to get an ARIA earlier so we can intervene? So this is my pitch for proteomics. So we've heard about Olink, and I'm going to talk about a different technology called NULISA by Alamar Biosciences. It's a similar type of technology. I would say that there was one key difference is that the multiplex that you measure with this technology includes phospho-tau 217, GFAP, and NFL. So it includes the biomarkers we know work very well, and you can build models around that. Me, as a skepticist, I like to know that, okay, just because it measures 217, is it a good 217 or is it a poor 217? So we've looked at this. So this is the Alamar in this multiplex, remember, so you're measuring it with 119 other proteins at the same time versus let's say the best in class, 217, and it's as good, if not better, at detecting AD pathology in these cohort of about 1,000 individuals. So the biomarkers within the panel say say what it does. So this gives us an opportunity now to do something very similar to what Charlotte has shown, and ideally you would do both proteomic platforms in the same patient so you can cross-validate targets, or both proteomic technologies probably don't have the same targets. If you can cross-validate results from orthogonal methods, it gives you a much more robust finding. So some of the case studies we've done, this is early tau deposition. So this is everyone that's amyloid positive but starting to develop tau. So what you can see for this technology is very, very nicely that phospho-tau are our greatest predictors, and that's what we'd expect. But you start to see a lot of other interesting biomarkers that are also increasing or decreasing in response to amyloid. This is in mild cognitive impairment, so those that have progressed from mild cognitive impairment to dementia within 2 years. Again, phospho-tau217 is our best predictor, but a whole host of other significant biomarkers that could be important and improve our prediction models. More needed tests, let's say for Lewy body dementia, and this is again in plasma, can you separate AD from Lewy body dementia? Phospho-tau217, of course, says that it's amyloid, but there's a lot of different biomarkers, again, significantly associating with those that can separate Lewy body disease and dementia in clinics. And again, as we've already spoken about inflammation, this is a much needed tool for us in every single aspect of Alzheimer's disease. So what we've done here is that we have the Alamar system here in CSF, separating those that have TSPO PET positive and negative. And here we have the plasma version. So a lot more markers in CSF associating with TSPO PET, as we'd expect. And there are some associating with in plasma, but there are some markers that cross-validate across plasma and CSF, suggesting that this marker is truly measuring at least a proportion of CNS-type inflammation. So these markers would be FAB-P3, which you knew about, ST3, YKL40, and GFAP. So to conclude, phospho-tau217 is a fantastic test for AD pathology in approaching clinical use. We need to make sure that the right test gets into clinical use and we use it appropriately and understand what it means. Phospho-tau217 most likely reflects amyloid pathology in the early parts of the disease, but then reflects tau, and that's probably why it makes it a better biomarker than 181 and 231, because it has this dual pathology association. It's not increased in other dementias, unless amyloid is present. It could have, if you have a different assay design, it could have non-specific binding. And this is just a development that we're going through blood biomarkers. If you think about how tau PET and amyloid PET started, they had traces that had non-specific binding. And this is just a process we're going through at the moment. But it can be increased in acute disorders. And phospho-tau217, as I say, is a fantastic diagnostic marker, but it explains a lot of variants in other solutions, but it's not the sole answer. We need to add biomarkers, and this kind of really replicates what's been said previous to me, to support treatments, to try and predict who's going to respond to such treatments, and of course co-pathologies that occur with Alzheimer's disease. This is the group in Gothenburg that have done all this work, and some key collaborators on the right. And as James says, I'm going to the Banner Institute, and I'm looking to take some people with me. So if you fancy doing some biomarker research in the desert, please come and speak to me. Thank you very much.
Thank you very much, Nick. Very informative talk. How close do you think we are to have a standardized reference range for phospho-tau for patients? And how did you decide the cutoff for your automated phospho-tau217 assay?
Yeah, so this is an important point. So you have to remember that an immunoassay is not an absolute quantification of phospho-tau in blood. It's drawn off a calibration curve. So each assay is going to give a different value. But those values are not necessarily meaningful because we don't actually know the molar level that they are. It's just read off a calibration curve. So there are starting efforts where you have a reference material, which is a known amount of phosphorylated tau, which each assay can calibrate themselves to. That is a lot of work. So we've only just got there with CSF. So that work is ongoing, but it's going to take time to have a kind of global cutoff. So what you will have in the next few years, and maybe Charlotte can comment later, is that you're going to have different cutoffs for every single assay, essentially. And that's what we had for CSF for many years. It's not really a problem. And each lab may have their own independent assay depending on their population. So that's something it's going to be. So in that study we just had there, it was essentially a 2 cutoff range. So we had a cutoff which had very high specificity and a very high sensitivity, and then had this, what we call intermediate range, where the assay isn't so accurate, and just acknowledge that these people would need further testing or tested later on. That was about 15%. Now, that 15% is quite interesting because if you look at the amyloid PET, they're also intermediate on the amyloid PET. So it's not necessarily the biomarker is inaccurate, it's actually reflecting that these people are going through a transition, actually. So, so whether this 2-state point cutoff is actually the right way to go, I think, is under debate, and whether you just have a single cutoff. with high specificity or sensitivity if you want rule in or rule out in your test. It's all depending on context of use. Clinical use and trial use have different meanings. So,
two quick questions. One, when you talked about the sources of variability, I was surprised that you didn't mention ethnicity or very old age, sort of 90 onwards. Can you comment on those? And then sort of going back to the geekery team, why is it that one epitope is better than others in terms of, what are the mechanisms of this?
Yep.
Is it just an assay thing, or is it something to do with the tau itself?
Yeah, both good questions. Ethnicity, I don't think we know the complete answer yet. These are ongoing studies, and it's just such a difficult thing to quantify. How do you define ethnicity? It's a difficult thing. And through what we've done, we don't see much difference there. But what you do see is a difference in the comorbidities, higher cardiovascular rate, which could affect the biomarkers and aspects like that. But simply, we just don't know the answer, which is why it's not there. The second thing is why are some epitopes better than others, and there's multiple aspects to this. The first one could be just better biomarker development. The field has really focused on producing very, very good phospho-tau217 markers because they thought that was a target to go. That could be one aspect, because we do see some 181 biomarkers from companies that are as good as 217. Another thing is that we can measure phospho-tau 181 and 231 in all of us without question. So there is quite a noisy baseline of these biomarkers where I would hope in most of us phospho-tau 217 levels would be almost undetectable. So it has a much larger dynamic range of movement when someone gets disease.
Thank you so much. I can see there's a few more questions coming, but to keep to time, I might ask if we can put those back to the general discussion. I'd like to thank Nick Ashton once again for a fantastic talk.
Thank you very much for the kind introduction and the invitation, of course. Well, I had the honor to speak in this session after a crew of 3 people that I thought I can't beat. So what can I do? And then, of course, the thing that came into my mind is the work that we are at present doing in characterizing aging using the proteome. So why age? Well, I think it was already introduced, the fact that age is the most important determinant for Alzheimer's disease. And even compared to ApoE, if you just consider the curve of the risk curve of disease over age, it's not only ApoE that is a sufficient cause of the disease, you have the age in order to get it. It's very different to be an ApoE44 carrier even and have developed the disease before the age of 40. So you need the combination there. So it's an important factor, and many of us working in genetics for a long time, and I'm thinking of the past that we have been working on geneticists and always wonder what is this factor in that aging process that is leading to this vulnerability of Alzheimer's disease increasing. So age is, of course, a factor. It's dependent on when you are born and what is your calendar age. But probably aging as such is the important underlying driver of the disease. So 2 persons with a similar calendar age or birth date can really have different biological age if we start looking at that. Now, that has been done already for quite a while, quite successfully, by looking at the DNA. So if you look at somebody's DNA and look at the methylation, so that is after birth you can change your DNA status by methylating, and that's usually susceptible to the environment, but also genetically determined. That is a very good marker to determine also how much of an aging process is ongoing. However, this DNA methylation has been relatively unsuccessful if you apply that in the setting of dementia. So the new opportunities we saw with the emerging opportunities in the biomarkers and the protein-based biomarkers is use the proteins as a readout of biological aging and characterizing people in the general population based on their protein profiles. So we were not the only one doing this, and actually in the start of this year or the end of last year, there was a fantastic paper coming out in Nature. And if you haven't read it yet, recommend read it at once, tonight or tomorrow if you go back home, because it's really a very nice paper who looked at organ-specific aging, and they looked really at different organs, not only the brain. And what they found is that the measurements that they created based on the organ-specific ages were also reflected in the blood. Now, actually, we always suspected that. It was not new, but it was an important factor that really gave a connector to one of the other mysteries that we always seen in epidemiological research, that there is really a very strong association between vascular pathology and your risk of dementia, and even of Alzheimer's disease in many studies. Now, if you look at it from the point of causality, the Mendelian randomization studies never really supported that, but we keep on seeing it. And one of the nice things of this study that was published in Nature in the end of last year is that it really suggested that the aging of the brain really was quite strongly associated with vascular aging. And that it showed an association with beta 181 independently, but also it was strongly a predictor of vascular aging, as was beta itself of disease. Now, whether that can be replicated, I can't give you the information yet, but it really showed that this trend was there amongst several studies. What it also did is linked aging with vascular calcification, the ECM, the extracellular matrix, as a mechanism, and synaptic protein shedding to cognitive decline. So this really complemented, because when this study came out, we were resubmitting our paper to Nature Medicine, which is now in press. We really tried to do the same thing, but independent of this organ-specific aging. So we really looked at the proteomic spectrum in blood and tried to make an estimator for biological age in the general population and link that to the risk of Alzheimer's disease and dementia. So we started out doing that, and I think the other study was a little bit ahead of us already, or study doesn't come out yet. But we started doing this in the general population, and we used on the one hand DPUK Biobank, and on the other hand the China KBRI Biobank, UKB and CKB, and of course these are two studies which are based in Oxford's Big Data Institute. If you look at the studies, they're a bit different. So on average, the people in the CKB in China are a little bit younger. It's depicted here. Looking at the age of death, to my surprise at least, the mean age of death is a little bit later in the China KBRI Biobank. What is clear is that the size of the study is a little bit different. I think the largest study is by far the UK Biobank. The CKB is much smaller, and that's also reflected in the number of patients what we see. In the CKB, we can study vascular diseases, but it's very difficult to study dementia and Alzheimer's disease because there's so few people at old age. They have more people at a younger age, around 40. All right, so how did we do the study? The study was actually very simple. We did this using machine learning, and to do a study in machine learning, you usually make a split sample set in which you have a training set where you just learn the computer how to estimate the age, and a testing set which you use to validate the findings. Didn't I overfit the model? We started out doing it with a combined set. So what we basically did, we took 70% of the people in UK Biobank with the randomly selected people, not the selected ones by industry, and split those from a 30% test set, and we did the same in CKB. So if you do that, you get a combined, for 2 studies, training set and a combined test set. But for the Nature Medicine paper, we really had to redo that in different ways. So we also had an alternative analysis to train the data purely in UK Biobank, which was easiest to do because it's the largest study, and predict only in the China KNU-KBRI Biobank. But we added another study because, of course, we worked with these 2 studies with very different ethnic background, but we were asked to have another study to replicate, and for that we used FinRisk. So that's a study in Finland. Again, a very different genetic makeup. So it doesn't matter what you do on the fly. If you worked with the Oling platform that was used in both UK Biobank and in the CKB, it's a little bit difficult to cross over data from one biobank to another without crossover samples. But luckily, we were able to develop together with the board an algorithm to do so by restandardizing the data set. So doing so, really, the findings are very similar. It doesn't matter how you split the data, how you combine them. We do find a biological clock. So how did we do the analysis? Machine learning, we used gradient boosting initially. We did the 5-fold cross validation. So you do it again and again and see whether you have the same finding. We selected the proteins with the Bruta feature, and we tried to maximize really for and capturing all the proteins that are necessary, and that made us end up with over 200 proteins. So what we did basically to estimate the predicted age is just regress out the age against the predicted age, and that was the first readout that we used across the different biobanks. So how well does it work? Well, if you look at the R-square, the R-squares are quite high. So the R-square is above 0.85 in the UK Biobank. It's also quite high in the relatively small CKB sample. And if you combine the sample, you end up with an R-square of 0.85. So what do we do next is just compare whether somebody shows evidence of accelerated aging. So that basically means that if you look at the line that is given here in red, that you are at the same age, you have evidence that you are older than you were to be given your calendar age or your birthday. And if you are below, you are a lucky person who aged less fast than expected on your birthday. So again, that was replicated. We find a similar set in the FinRisk, but we also find that it doesn't matter which technique we use. So instead of gradient boosting, we were asked to use a classic net or a neural network. We find very similar results. The only thing that we found for the neural network, that it was a really bit more confused if you were on the edges. So if there is no data on people who are younger than the calendar age, which is the case 40 years and 70 years for the UK Biobank, the neural network was the one who performed worse on that part. So how much a difference does this biological age clock make? Well, quite. So if you look at the people with a similar age, you can be 10 years younger or 10 years older. Of course, these are the most rare people. But the difference is about 20 years between the top agers and the least lucky people who aged least. We see that quite similar effects in cross-ethnic groups in the UK Biobank. Of course, the Asians and the mixed and the blacks are quite small groups of about 10,000 in UK Biobank. And in China, the CKB is really designed over different populations, so that really goes all over the empire, so that is quite different ethnic groups also involved, and we see quite similar effects of the estimate of the age. Now, do we give new information on what was there already? And of course, the most important clock that is out there is based on DNA methylation. And what you see is that basically the overlap between the methylation clock and the proteomics clock that we find is quite limited. And how to think about that is that if you think about what happens with methylation, you just get over your life course all kind of exposures. That may be your smoking history, but it may also air pollution, but it may also alcohol, your diet, lifestyle, etc. And that gives you an imprinting in the DNA, but that is random. So it will go over the full genome. So it may be occurring in coding regions or non-coding regions that don't code for any protein at all. So I think what we are hypothesizing that there is an overlap, and the overlap that we probably see is that there is methylation in these coding regions. If we compare our finding with other proteins clock, we also see a quite random pattern that we really have been very much targeted that we capture as much proteins as possible. So we have a lot of proteins that others haven't reported yet. If we try to reduce our proteins, we really can narrow it down to 30 proteins that are the drivers of all association. And again, these are showing quite a limited overlap with these other proteomic clocks that have been published. So what are we picking up? Well, of course, if you look at the Olink platform that we used for these data analysis, we have limited information. There's no information on tau. Nick already mentioned this. That is the big missing factor for Alzheimer's disease. But if you look at the other proteins that are really important are NFL and GFAP that we of course pick up, but we also pick up a lot of cytokines, which is what we expect. I think if you work in aging research, we often think of aging as inflammation as the major factor. The ECM really pops up as an important pathway, and we also pick up other pathways that are known to be involved in aging, like the IGF-1 signaling pathway that across all animal species is very important in aging. So what about the biomarkers and the risk factors of aging? Well, we determined that cross-sectionally. We used the UK Biobank for that. And that has, of course, a wealth of information. And we did that in the full sample. So these are including people with diseases, but also in a sample that we were very strict saying they don't have any active disease yet. So if you take the full sample, you see that aging really is very strongly associated with kidney function. We all know that. But also liver function is an important driver. And of course, inflammation pops up, but also a very interesting one is the telomere length that we see as expected, that your telomeres are one of the clocks that are ticking in terms of aging, that if your telomeres shorten, that is usually a sign that you did a lot of mitosis, and therefore you have shortened telomere lengths and are on the way to aging and age-related disorders. IGF-1 also very important. In the disease-free, these two later ones pop also, but also you clearly see that the kidney function and the liver function is also very important. If you look at the more clinical orientated factors, what is an important factor, of course, is that how well do you feel yourself? I think that is one of the first findings in the UK Biobank, that is such an important predictor of age-related diseases, but also being slow in general, rating yourself very much aging in your face is really very strongly associated with this aging measurement. The long sleeping isn't associated. But more important, if we think about Alzheimer's disease, that is relevant for today, we do find an association with intelligence and the reaction time. Where is that? I can't see it very well. These are, of course, the outcomes that are mostly risk factors, but we also can focus on mortality and morbidity. And the first part that we look at is really looking at mortality overall, but also looking at the risk of diseases over time. So in blue, you find what is the risk of the disease by age for the top 10% people who age the most accelerated compared to the people with the same birthday. In orange, you find the people who are in the medians, and the lowest in green are the people who age slower. So they really are the most successful agers. So the highest risk, of course, is seen for people with all-cause mortality, and that is logical because aging will give you all kinds of diseases, but we also see very strong effect for type 2 diabetes, ischemic heart disease, stroke, and of course kidney disease, liver disease, and lung diseases. If you look at the UKB, that's the largest sample, but the much smaller sample, CKB, also shows the same findings for these vascular outcomes, but unluckily they don't have available any data on dementia in sufficient numbers. So what do we see for Alzheimer's disease and Parkinson's disease? Well, if you look at all dementias, similar to Alzheimer's disease, you see the top 10%, of course, ages more strongly than anyone else. And you see that the risk in those who are on the median, so the average, what everybody on average has in terms of aging, is very comparable to the lowest 10%. So those up on the age of 65 have an over 30% of probability of developing Alzheimer's disease during follow-up. But if you look at Alzheimer's disease specifically, you quite see the same distribution, although the overall risk of Alzheimer's disease is much lower if you look at the absolute risk. But that has a reason, that is a little bit of bias in the UK Biobank, that for half of the patients with Alzheimer's disease, the diagnosis is unknown. Also for Parkinson's disease, you do see the same trend emerging. So again, the top 10% of the people who age have the highest risk of Alzheimer's disease. So how independent are these findings? Are they really explained, and that is what we started out with, by these vascular risk factors or other risk factors that we know of that are involved in the disease, and that may involve the education or the deprivation index, the physical activity, your smoking status, how healthy are you, but also, of course, BMI and hypertension are major drivers of dementia as well as cardiovascular disease. Now, whatever we do, whether we adjust just for age and sex only in the first model or in advanced models, including more and more risk factors for cardiovascular disease, what we do see is that these effects of aging just prevail. So what we're picking up is some sort of a measurement on how accelerated the aging process is, independent also of this vascular pathology. So what do we think of this? How can we use this? Can we use this in trials? Well, maybe yes, if you think about that. If you think about including people around the age of 65 years in trials, if you select the people from the lowest 10% of the agers or the median, you may improve your probability to have a positive hit if you just take the top 10%. So if you looked at the accelerated aging, persons, you probably have a much higher risk of developing dementia. So you may think about including these people in trials of dementia or Alzheimer's disease specifically. So what are we missing? In the UK Biobank, and this is one of the challenges that we're working on as a follow-up of the paper, is we cannot access the association with p-tau, but we do see strong effects, and it really we're talking about the most strong effects that we are seeing, even if you adjust for age itself. However, there's also a downside, that is, if you are selecting in a trial those accelerated aging, you're getting also a lot of multimorbidity. And with that, we mean that if you think about the number of the diseases, that the people who really have one disease in the top 10% of accelerated aging is relatively small. So thinking about trials, you could also argue the other way around, that if you include people in a trial with a high biological aging score, you also are including particularly people with multimorbidity on the scales of cardiovascular disease and also other diseases, including diabetes. Whether that is a problem, I was quite surprised that that problem also emerges already by the age of 45, 50 years. So it means that this aging process really kicks in quite early, and not only in those 50 to 65 years. So the take home, if you look at the UK Biobank, I think the top 10% of the accelerated aging people are really showing the dramatically divergent age-specific risk curves for both mortality and morbidity, and that really leads to multimorbidity that you see in these patients. Alzheimer's disease and dementia really stands out. If you look at the relative risk compared to the no, because of course Alzheimer's disease starts later in life, then most of the vascular pathology, it really starts out as the most associated, strongly associated with aging. And if you look at the accelerated aging, you get not only those who are at high or low risk of Alzheimer's disease, but also of multimorbidity. That was my story, with the acknowledgement, of course, to people who did the work. Ashton Argentieri, that is a postdoc in the lab that really did the work with Sihao Zhao and Najaf Amin, and many thanks to the CKB and the Department of Psychiatry. Thank you very much for your attention.
So in my flash talk, I'm going to be focusing on neurogenesis-centred biological susceptibilities for dementia and the important role that diet could play in the trajectory of this condition. So to give you a very brief introduction, we know from the literature that diet can indeed modify future dementia risk. And the way in which we think this could be operating is via the food metabolome, through the release of endogenous factors that get into the blood, cross the blood-brain barrier, and then ultimately interact with the brain. Now, a key brain region relevant to this is the hippocampus, and the reason for this is that the hippocampus is one brain region in which an important biological process occurs called neurogenesis. And neurogenesis really is crucial in this context, largely because neurogenesis can indeed be modified by diet, but also more importantly, the process has also been associated with the pathogenesis of dementia. However, currently it is actually impossible to test the effects of neurogenesis and then the effects of diet on neurogenesis in live humans. So in an attempt to address this, we in the Thurrott lab developed an in vitro cellular assay that enables us to generate proxy readouts of the neurogenic process from participant serum. Essentially, this involves simply adding serum from each participant to hippocampal progenitor cells and then measuring the effect this has on the neurogenic process. So using this approach and serum samples and data from a clinical cohort, the 3-city cohort, which is a longitudinal demential cohort consisting of French elder age subjects 65 years and older, we subsequently tested the association between neurogenesis 12 years prior to condition onset and the development of future dementia. What we found was not only was neurogenesis at baseline associated with the development of future dementia, but that also this was characterized by specific neurogenesis readouts that then mapped onto specific subtypes of dementia. So given this, we then subsequently characterized all participants in our cohort according to whether they had high or low levels of these specific neurogenesis readouts, essentially assigning a susceptibility status to each one of our participants. We then wanted to determine whether or not diet at baseline could be interacting with the susceptibility indicator and potentially altering your dementia outcomes 12 years later. To do this, we ran a series of multivariable adjusted logistic regression analyses to essentially test the interaction between various nutritional and dietary markers, neurogenesis susceptibility, and then ultimately the development of dementia outcomes 12 years later. So what did we find? Well, essentially, we found that increased meat consumption, reduced poultry consumption, and higher plasma levels of lycopene, which is a carotenoid biomarker, all significantly were associated with the development of Alzheimer's disease 12 years later. But crucially, this was only in individuals that also had a neurogenesis susceptibility at baseline, as indicated by the red bars in the plots. We also found, and again, only for those with this neurogenesis-centred susceptibility, that increased vitamin D consumption was significantly associated with individuals that went on to develop vascular and other types of dementia 12 years later. So altogether, we believe that diet could potentially alter the risk trajectories for dementia outcomes, but that ultimately this may be operating for a specific subset of individuals, particularly those that already have a susceptibility, and in this case, a neurogenesis-centred one. However, future work really is needed to try to understand how all this may be operating. So thank you very much for listening. Happy to take any questions. And of course, if you do need any more detail, you can catch me at my poster session. Thank you.
So good on timing. I think we do have time. If there were one or two questions, we would have time. Send it in the back.
So actually, it's a little bit counterintuitive. So vitamin D is characteristically thought to be protective in dementia cohorts. And so what we're seeing actually, at least early on, is that it's more vitamin D is associated with an increased risk. But I think the thing to point out is that we don't really know what this means, because what we're capturing here is something that is happening very, very early on in the trajectory. And so while we don't know, we speculate that it could potentially be tapping into some compensatory mechanisms or changes in behavior that are facilitated by these earlier changes in neurogenesis. But we don't know.
I just thought, recently there's been massive prescriptions of vitamin D across the world.
I know.
And I don't know whether it will result in increased dementia rates later on. Thank you.
Thank you. Thank you very much.
Thank you.
So just a quick background, DLB is the second most common neurodegenerative dementia after Alzheimer's disease. And in addition to the neuropathological hallmarks of Lewy bodies, coexisting Alzheimer's disease such as amyloid plaques, tau tangles, and more recently microglial activation are all increasingly implicated in the pathogenesis of DLB. And this is one of the reasons why there is an increasing focus on identifying some of the neuroimaging MRI biomarkers that could be sensitive to these underlying pathological processes in DLB. And among some of these emerging MRI techniques, free water mapping using diffusion MRI is a very exciting technique because it can separate out the diffusion in the brain tissue from the extracellular space. And elevated free water is typically associated with aging, cognitive impairment, the transition from MCI to AD, and Alzheimer's disease itself. And it tends to be interpreted as being related to neuroinflammation processes in the literature. However, despite very common interpretations of free water as a proxy of neuroinflammation, very little has been done to directly look at the in vivo relationships between free water MRI and PK PET imaging of neuroinflammation in vivo. So in this study from the Nimrod project in Cambridge, we included 16 healthy controls, 25 DLBs, and all of these participants underwent PK PET to measure neuroinflammation, as well as diffusion MRI to measure free water levels. Now, if you're interested to know more about how we actually derive the free water values from the diffusion MRI datasets, you're more than welcome to visit me at the poster for more technical discussion. So the first question is global free water in the gray matter increase in the DLB groups compared to the healthy controls? So as you can see from the violin plots here, compared to the controls, the gray matter free water, at least globally, was significantly increased in the DLB group. So this is very largely consistent with the previous studies that have been done in Alzheimer's disease and other neurodegenerative conditions as well. However, the analysis that I just showed you is only presenting a very global picture, so it is unable to actually pinpoint the exact locations in the brain where increased free water is most prominent. And we were also curious to know whether increased free water is independent to that of gray matter volume loss or atrophy. So in the next analysis, we ran multiple linear regression models to compare regional free water across the gray matter. And we found that increase in free water was most prominent in the temporal, parietal, as well as the occipital cortices in DLB. So all these color regions here on the brain maps are areas where increased free water survived multiple adjustments for correction, as well as regional gray matter volume. And the fact that this increase in free water survived adjusting for regional gray matter volume is quite significant because it suggests that free water may reveal disease-related effects over and beyond that of conventional MRI techniques such as structural MRI. Now, finally, what is the relationship between PET imaging of neuroinflammation and free water? So to do this, we ran linear mixed effects modeling, and we saw a highly significant negative correlation between PK PET on the x-axis and free water on the y-axis. So in other words, greater neuroinflammation in the brain was actually associated with lower free water values in the gray matter. And what was also quite interesting is that these significant associations were found separately in both the controls in green and the DLBs in red. So what are some of the take-home messages? So the increased free water was independent of gray matter volume loss, suggesting that free water may be sensitive to these microstructural alterations over and beyond atrophy. Now, the negative associations between PK PET and free water was somewhat less straightforward to interpret, although some of the plausible mechanisms for this correlation may involve the microglial infiltration into the extracellular space, therefore restricting the water mobility and finally manifesting as reduced free water. Although these are just cross-sectional correlations, so we definitely do need to do longitudinal studies to confirm these results and also to determine whether baseline free water MRI may actually be useful in terms of predicting progression from prodromal to probable DLB cases. And finally, one of the last area for future research is to correlate free water values with all the exciting blood biomarkers that we have heard so much today about. And with that, I end my flash talk. I would like to thank all these people who have been working on this project and also the funders that have made this study possible. Thank you.
We should come up with a question in 2 stages. So we'd like to elicit your views, having heard the sessions this afternoon, really on the first question around what would describe a successful academic-industrial partnership. We've actually heard mainly from academic speakers this afternoon, but throughout the talks it's clear about their interactions and dependency for the success of the work on industrial partnerships and technology development and looking ahead to deployment of biomarkers. So you may also, through the lunch breaks and tea breaks, have spoken to people from a different sector from the one you naturally inhabit, so academic to clinical to industrial or third sector. Do have a think about what you feel makes a successful academic to industry partnership. I think that's really valuable. Thank you. I think it's really exciting to see that, the sort of common ground we have here in the room across people of many different backgrounds, career stages, sectors in the dementia research community. Yes, as we all need to build on and work at trust and maintain and build trust for that success. We will take that forward, we'll take that offline and then move to the second question. I think that many of these are the issues in terms we've heard from our speakers today as trying to look towards accuracy, prediction, reliability. Thank you for joining back up on the stage here. We've had a few questions as we go, But would anyone like to ask any general questions or any questions for any of the panel members back over what we've heard earlier in the afternoon? Gentleman in the middle there.
Thank you for the fantastic presentation today afternoon. My question is about, I work in a memory clinic. So in our regular practice, we get an overlap of people with depression, other, factors which are associated. I'm not saying they are all dementia related, but could be normal aging and other factors like functional cognitive disorders and various others. Are the biomarkers quite specific for conditions we already mentioned? Or, yeah, just whether we will be able to differentiate. And one more question, I think, with the inflammatory biomarkers, I just wanted to check delirium, is that something which we will be able to kind of, find out through using the biomarkers? Thank you.
Thank you. I don't know whether Charlotte might want to lead on that.
Well, within a memory clinic setting, the amyloid and tau biomarkers, they are very specific. So they will guide you. But it doesn't mean that there is no other pathology ongoing. So that's important to realize. It's not only, but it's the likely explanation of the cognitive decline once you're amyloid positive. And Peter 217 within a memory clinic is very specific. I think Nick can also agree to that. And the examples that he showed in ALS or newborns and TBI, it's clear that it's not completely specific. So we have to tone down that statement maybe a little bit. But within a memory clinic setting, it is. And I think that's very relevant. And but with respect to neurofilament light, for example, that's more general neurodegeneration biomarker. That's the beauty of it because it's very sensitive towards changes. For example, if there are in multiple sclerosis, we observe that if there is if we stop treatment, if people are very long on treatment, and when they're older, we thought we could stop. But then neurofilament light was quite specific for detecting novel disease activity because many people had, again, disease activity, so it was not a good idea to stop with that treatment. But it looked very specific. So within that setting, it can be a very good biomarker to detect novel disease activity. But on the other hand, if for a specific diagnostic tool, it's a-specific. But in some diseases, the levels are sky high, and then you can use them, and along with other measurements.
Thank you. Any other questions from the floor? Lady in the middle end.
Hi. I just wanted to revisit something that's been touched on a few times today, so the equity, diversity and inclusion issue. There was a comment made earlier with regards to inclusivity of people of different ethnicities, which I found quite tricky, because the underlying tone of it, for me at least, was that we can't possibly do that because we can't define those ethnicities. We're not doing a great job for anyone who isn't white, never mind specifics of certain ethnicities. So I wondered if you could comment a bit further on that with regards to your opinions about where we're at with that sort of thing, because I don't think not being able to get into the nuance of specific ethnicities is what the question is. I think the question is, what are we doing to look at people who are not white?
And that's a really important issue. I don't know this side might lead on that.
Yeah, so I guess we've both done work in this area. So all of the cutoffs that we have done and presented have been generally done in a Caucasian European population or white American population. I think that what is ongoing a lot in the United States, here in Europe, is that we need to know if these cutoffs are the same in different populations. And in order to do that, we need gold standards. So we need CSF biomarkers and PET biomarkers to understand if the blood biomarkers are the same. So that's kind of the engagement where we need to revisit. And I know there are a lot of efforts to do that. to kind of re-establish whether these cutoffs are the same, and if they are different, what is driving that difference. I can't remember the specific names of the study, but there is a lot of ongoing efforts, particularly in the United States, in order to do that. Charlotte, do you have any other further comments?
Yeah, so it appears that plasma p-tau levels are somewhat higher. But for me, also supported by the results of Cornelia, I cannot really believe that the biology is substantially different. So there could be other causes for that. But yeah, okay, the results are there, so we have to understand them. Yeah, and in the Netherlands, we're not allowed, or we were not allowed, but it's still the case, to monitor ethnicity. So we will never know. I can see from names that they are not Dutch, but yeah. So, but that's just a guess. So it hampers our research. The nice thing about our collaboration with Rotterdam, in our prospective cantate study, in the real world study, is they have a special outpatient clinic for underrepresented groups, so minorities, but in Rotterdam it's not a minority, a majority. And yeah, so put special efforts also in translation and so on. And they are very happy with our plasma biomarkers, because sometimes the cognitive tests are difficult to do. Yeah, so from there we will get a more mixed population, because they are also participating in our studies.
I also think it's important to. I mean, we've just published a paper recently with a group in Singapore, and in Singapore there's a much higher level of vascular dementia or vascular vision. You see that you see that reflected in the biomarker of that population, in neurofilament light and GFAP. I also think it's important that now the whole idea of a blood biomarker is that it's meant to be simple and accessible. So therefore, groups that are trying to investigate their own population should be able to do this themselves and not have to send it to Gothenburg to me in order to do it. And that research should be able to be done within their own institution with their own questions.
Yeah, it's also a matter of participation in research and trust in institutions like academic hospitals. That's why people did not participate in research. But yeah, it's an important issue.
I think David Bennett was trying to come in from the back.
Oh.
Hello. Oh, there we go. I just want to say that this is a really, really important issue. I can tell you that this has led to a number of very agonizing, long, drawn out, in the public arguments about some new criteria that have come out of the United States which have not been adopted in Europe. But there's little question that ApoE is under positive selection in Africa. The population distributions of ApoE are very different. Their effects clinically are very different. All right, and one would wonder whether that would follow through with the biomarkers, be they from the brain, the CSF, the PET, or the blood. Okay, and there's simply not enough data to know what to do with this stuff. All right, and there's no solution other than to get the data. All right, and there's other genomic differences that are enriched in Africans, and we're going to see more of this come out, because through the ADSP and the ADGC and other efforts, we're learning more and more about what's in these populations and how they differ and what's under positive selection. And then you're going to get all these weird admixtures across all of these countries where all of these groups have mixed for 500 years. So I do think that it would be great if Europe came together, like the United States hasn't quite managed, and help generate these data and make them available for everybody, so we know what to do with them.
Thank you. And I wonder whether Cornelia wanted to add anything to that.
Yes, so I think we were also a bit amazed at how similar the patterns were in the China KNU-KBRI Biobank and the UK Biobank. Not everybody may have realized that these two biobanks were developed synchronously, so together, hand in hand. What I think is important also, David, is that what you do with the proteomics, I think in most cases, look at very much downstream effects of the genes. And although the causes of the disease, the genes may be different, as long as you don't have a unique causal pathway that is quite unique for that genetic makeup, you start seeing the same endpoints. We did the same track with chronological aging, for instance, in the setting with the effect of smoking and diseases, but also other outcomes. And you see that it doesn't matter whether you smoke in China or in the UK, the effect of the smoking on the protein profiles and the effect on the diseases is quite similar. So I think that is the difference with genetics. All these genetic makeups, unless they give a very specific pathway, they go to the downstream effects that are very similar.
Thank you. And Do-Geun, you studied the Korean population. Are the omics signatures you see there different from what you read in the Western literature?
Yeah, it's very hard to say that, but at the same time, as you pointed out, the instances of dementia and neurodegenerative in South Korea is ever increasing, and we're worrying about it very dramatically. And at the same time, government is trying to focus on it to see the effects from the South Korean population. One of the key questions should be whether South Korean, as you already pointed out, ethnicity change differences in between the Korean and the Chinese people and the European people might be a little bit different, and at such a little differences might be having some of the question whether the therapeutic approaches for those Korean populations should be the same for European people or the American people or or some other ethnic population as well. So I think it should be really important for us to, I mean, I'm not at the stage that I can say that, but personally, I think it'd be very important for the academic position or the government position to have a, to have inter-ethnic differences, including the Asian people as well in this context, because there are some reports that the Asian people have a different susceptibility, especially for the high-fat diet, because at the same time, the ApoE responsiveness is a little different to that of the Caucasian people. So such things should be very important topics in the long run to be considered in the later on. That's, yeah.
Thank you. I think this is an issue which will be very live and relevant through tomorrow's sessions on trial design and big data as well. It's a current theme. I wonder whether there's any other questions from the floor. Yes, right at the back. And
thank you all very much. Megan Holland from the University of Cambridge. Professor Teunissen touched on the exciting opportunity or exciting prospect of a compound score quite similar to the Framingham score for predicting dementia risk. And I just wondered, how do we implement a biomarker like p-tau that has these sporadic spikes when measured, say, on day 1 and day 10? How do we allow for these kind of heterogeneous biomarker levels? Do we need an aggregate score? Do we measure a patient's p-tau over a week or a month and then put that into a score system? I'd be interested in your thoughts. Thank you.
Yeah, so these cases are rare. So this was just an illustration of that there are some things that affect these assays we don't know. So first of all, it's rare. Second of all, it's not uncommon to reconfirm a positive test in clinical chemistry. So this would be a standard routine anyway, and it would be caught out in that regard. So it's an illustration of there's some things we don't know about these assays, but I think it will be resolved in the normal way that we deal with positive tests in clinical chemistry.
thank you. And there was a question in the back middle, so yes.
Hi, a quick question, probably mostly directed at Nick and at James. How far are we away from using phospho-tau 217 in research, in the clinic, and from a regulatory perspective? Let's say I'm a drug developer who wants to develop a new Alzheimer's drug. What biomarkers should I be using to define my population at this point? Do I still have to go to PET? Can I be using CSF-A beta? Or can I be using phospho-tau 217 in the blood?
you want to have a first go at that? I have my thoughts.
Yeah, okay, I have a first go. I think Charlotte should say something as well. I would say in research, it's now becoming the accepted biomarker to have. So talking purely from an academic point of view, if you're trying to publish a paper on blood biomarkers and you don't have phospho-tau217, you're going to have a difficult ride. So coming from that aspect, you know, in clinical chemistry in Sweden, where I am now, we are implementing this test now as a test that a clinician can choose to use if they want. We have a different regulatory system. It can be approved by a clinical chemistry board of examiners. So that test will be available, just like neurofilament light is already available, has been for several years. And I'll let you speak about the UK. We know that a lot of these drug trials, like the donanemab trial, have used phospho-tau217 as one of their inclusion tools for these trials. So they are trusted on that level. And from the data that I've seen from conferences, that seemed to have gone very well as an inclusion criteria for these trials.
No, I think that's not correct, because that's post hoc maybe, but they use tau PET as an inclusion criterion in the donanemab trial, because the tests were not that advanced by then. But it's a fact that many drug companies are now using they're taking the step towards pre-screening using the plasma biomarker tests. Also, I think supported by the appropriate use criteria, but also now the revised criteria. I think that's it's almost a reality. And I expect that in the Netherlands, we're dependent on the certification by the IVDR CE mark of those tests, whether we can use them in the clinic. But we're now prospectively testing, as I indicated, in our memory clinic. And I expect that we will test for half a year more, because first we started with 181 and now we're testing with 217. And then with enough information, we can just start like that in all our patients. So I think we're almost there, and I hope that there will also be this certification that's needed for clinical decision making. That's in parallel with our process or research reports. So I hope by the end of this year.
Yes, I think you were right there to separate the role in research and clinical practice. I think they're two very different domains. I think already the 217 is prominent for screening as an entry to research, but it may not be, and probably isn't a tool you'd want for monitoring response to therapy, and you may need other biomarkers later in the trial. For healthcare, I think I would see it as having an important role to augment and assist a proper memory clinic assessment, not to substitute it. It cannot be a substitute for a conversation, a history, looking for other treatable factors, proper examination, and actually other investigation to rule out things other than Alzheimer's. That may include scanning. So it can enrich a memory clinic assessment, but you should be asking yourself as the clinician, how would a positive result help me? Or how would a negative result help me? And if you don't know the answer to that question, you shouldn't do the test, as for any other medical test. Don't do it blindly and hope it'll help you, or you'll get into more difficulty. You need to know how will the answer help you with this person in front of you in their particular situation.
And I want to stress that even in the revised criteria, we're not propagating that it should be used outside the clinical setting. So first the clinical question, and then in replacement of CSF or PET, or if you don't do it because there was anticoagulation therapies or there's another reason for not doing CSF analysis or PET, then you can use the plasma biomarkers. But it's not a screening for population screening or testing in unaffected individuals, always in the clinical setting.
And I think on that note, I think we need to call it to a close for today. I'd like to thank all my speakers from earlier in the session today. Thank you.
Thank you.
And I believe we have a reception. Oh, thank you. Yes, come in.
What a fantastic set of talks and discussion this afternoon. I'm Iain Chessel from AstraZeneca. I'm slightly alarmed by the aging and diet talks. I think I'm probably doomed. But on to the arguably the most important agenda item of the day, which is a drinks reception. So this has been kindly sponsored by Acrivia Health, and Ben's going to come up in a moment to say a few words. I was really pleased actually by the word cloud. I was going to say a few words about collaboration between industry and academia, but the word cloud actually did most of my work for me. But I think it probably is worth saying that this collaborative environment that we're in here and within DPUK is really essential for us to succeed. You know, industry is really, really good at turning out assets. Academia is really, really good at basic mechanistic research, and actually industry is not very good at that. So working together collaboratively, you know, we can make such a bigger difference than working independently. And I would definitely say that sort of one depends on the other. And DPUK actually is a great example of that. It's a high trust network with collaboration between industry and academia. It's very open. It allows us to communicate and figure out what we need to do next. So it's a really, really good example. That's my plug for DPUK. With that, though, let me ask Ben to come up and say a few words before we imbibe a few aging drinks.
Thank you very much. So I'll try and be quick because I'm standing between you and the liquid refreshment. So most importantly, huge thanks to John, Simon, James, Sarah, the whole DPUK team for putting on such an excellent first day. I think reflecting on what we've seen, I'm always minded of John's opening statements when coming to the end of these days, that, as you've said, DPUK is a vehicle for collaboration between academia, industry, healthcare, and the third sector. I was reflecting, I think this kind of cross-sector collaboration is rapidly becoming not just a nice way to accelerate research, but actually a fundamental necessity. It's very exciting to see all the recent progress in dementia research, but with this progress, we're also seeing an explosion in complexity. This is only our second year at DPUK, but in that short time, we've seen this huge expansion in the data sources, in the data modalities, in the analytic methods that need to be used to kind of grapple with the questions that we're all addressing. And this complexity makes it pretty much impossible for any one researcher, any single institution even you know, institutions with a lot of resources to kind of do it all. At Acrivia Health, we've had some recent direct experience of this. For those of you who don't know who we are, we curate access to a large clinical phenotypic data set, so anonymized electronic health record data for around 5 million patients in secondary care psychiatry in England and Wales. And really excitingly, we're now beginning to expand that data set beyond purely phenotypic data to include genomic, biomarker, proteomic, and iPSC data through a program where we're recruiting 50,000 patients from our data set. When we began this program, I was speaking to one of our founders, Simon Lovestone, and asking, should I start hiring bioinformaticians, genomics experts to try and analyze all this data? And he quite rightly said, no, there's no possible way that I would be able to replicate the level of expertise, specialism needed to deal with that side of the data set in our small team. We should focus on our expertise, our specialism, which is on that phenotypic side, and build collaborations, find partners, bringing everyone together around the data set. I think the amazing research we've seen presented here today perfectly illustrates the previous comment that John and the team with DPUK have created a great forum for building those kind of collaborations. I noticed one of the stands outside says, right person, right place, right project. So, with that in mind, I'd like to invite you all to head out to the drinks reception, because in my experience, places with liquid refreshment are often the right place to find the right people for the right project. So, thank you very much and enjoy the drinks.